Assalamualaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you to online meeting on recent developments in drug discovery and medicinal chemistry. This meeting is organized by Comstech in collaboration with HEJ Research Institute of Chemistry, International Center for Chemical and Biological Sciences, University of Karachi. First of all, let me thank our distinguished speakers, our organizers, as well as the participants for attending this online meeting. And we will begin today with the recitation from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah. This online meeting is organized to discuss design, various aspects of design and development of anti-infective agents by using medicinal chemistry approaches, along with discussion on latest and outstanding developments in the field of medicinal chemistry and rational drug design. Some of the topics that would be discussed in this online meeting include medicinal chemistry and drug approval processes, drug targets, pharmacokinetics, and metabolism, along with some of the leading discoveries in the field of drug discovery and design. For this online meeting, we have around 700 participants registered with us, and these participants are coming from various parts of the world. We have representation from 15 countries like Nepal, Sri Lanka, Malaysia, Sudan, Nigeria, Oman, Pakistan, Gabon, Cameroon, Mexico. And we hope that this online meeting will be fruitful and engaging session for everyone here, including our distinguished speakers as well. And with this, I would request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCPS, for his welcome and keynote address. Over to you, sir.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning and good afternoon, colleagues in different time zones. Hazima, thank you very much for briefly introducing this very important event, which we are organizing in continuation of lots of activities which Comstech is conducting since last 1.5 years, largely related to healthcare research. And this is one important event which fits well the overall theme of capacity building. I'd like to welcome distinguished speakers from various countries, from China to Turkey, from USA, and other distinguished colleagues from different countries, including Pakistani scholars. And with this program is certainly an extremely enriching experience. And participant, as you mentioned, from 15 countries, truly an international event, not only within the OIC countries, but also beyond. And that represent the universality of science and technology and the need of spreading the message of science beyond borders. Now, comstech, which is Ministerial Standing Committee for Science and Technology, Ministers of Science and Technology and Higher Education of 57 member states in OIC region, is responsible of conducting many activities, including it serves as an observatory of science and technology innovation development in member states. It carries out lots of joint projects and capacity building initiatives, and it is also responsible of developing technology. In the context of technology, we consider capacity building as an extremely important element because only through capacity building we develop this capacity to absorb technology. Once we absorb technology, then we use it and then eventually master the technology. And medicinal chemistry is no different. Topic is extremely important, as mentioned by Khazima. Uh, Medicinal chemistry has been a very important subject throughout the history. It started from the development of drugs from natural product. And even at that time also, medicinal chemistry played an extremely important role in improving the pharmacokinetic properties of bioactive natural products which emerge as medicines. Medicinal chemistry has evolved over the year. So it was initially developed as a tool for generating tremendous quantities of chemical space, expanding chemical space for high throughput screening. So technologies such as combinatorial chemistry and high throughput screenings were put together to use the new space which medicinal chemistry has generated for phenotypic screening. The emphasis has been largely on hit and trial, and by using enabling technology, there were lots of exciting discoveries were made. But then medicinal chemistry has actually moved forward. It has acquired a new shape and a new magnitude. Medicinal chemistry has emerged as a science of known. Instead of science which was discovering unknown, medicinal chemistry has developed at the interface of chemistry and biology and emerged as truly interdisciplinary science in which uh, organic synthetic chemistry being the core discipline which was required with the competency of biochemistry and collaboration with pharmacology, toxicology, computational chemistry that has emerged as a science uh, in which the newly discovered molecular etiologies of diseases were used and employed. With the advent of modern science and technology, we now understand diseases at a molecular level, not all, but several of them. And these molecular targets become the main emphasis of medicinal chemistry because here medicinal chemists who are the architect of molecules start looking into the biological targets of different diseases, drug targets, and start designing molecules. That become an extremely exciting field because here you are not only doing any random large scale chemical space generation to phenotypic screening with high throughput format, but in fact, a lot of science went in and medicinal chemists start developing nuts and bolts of what is required to be complementary structure for drug discovery and development. That was a major shift, and I consider that as a historical turn into the science of medicinal chemistry. because today medicinal chemistry is largely based on target, and it based on designing complementarity of structures which fits well with molecular drug targets. That has given medicinal chemistry a new face and at the same time a new constituency and a new rationale. Uh, this has been seen during the pandemic. We have seen that COVID-19 pandemic has triggered and accelerated scientific development throughout the world. While molecular biologists and vaccinologists and virologists were busy in developing vaccines and biologicals, medicinal chemists, along with their colleagues in pharmacology, computational sciences, and other disciplines, were busy in developing therapeutics. And to the last count, according to the World Health Organization, there were 650 drug candidates which are in the pipeline of development. They are in different phases of development. So within a year and two, medicinal chemists were able to architect new entities which could treat COVID-19, which I feel to be the major solution of COVID-19. Vaccine is there to stop the spread of the pandemic. But when it comes to the treatment, then we need to have new therapeutics. At the same time, medicinal chemist has repurposed the old drugs. And there are a large number of old antivirals and anti-inflammatory drugs which were clinically tried. Medicinal chemists played an extremely important role along with the clinicians in solidarity clinical trial of the World Health Organization. And as a result of it, they were able to bring lots of new molecular entities. I don't really see a long time before we will be able to uh, see over-the-counter drugs for COVID-19. But this is not only COVID-19, which has, of course, highlighted the importance of the capacity in medicinal chemistry, because pandemic is not the last one. And pandemics would be coming even more frequently, and we would always need medicinal chemists to quickly come forward with new molecular entities for the treatment. There are diseases all the way through. Everywhere we have diseases of different kinds. Developing world, where most of the Muslim countries are located, is inflicted with triple disease burden. So we have huge communicable diseases, so-called infectious diseases of all kinds, including tropical diseases, and the non-communicable diseases resulting from overnutrition to malnutrition to undernutrition, which may cause a whole range of different diseases such as cardiovascular diseases or metabolic disorders of diabetes, but also at the same time cancers and others. So these lifestyle diseases are also very, very common along with infections and tropical diseases. Along with this, we also have another very unique disease burden, which is related to genetic disorders and reproductive health issues. Neonatal death is very common. Child mortality is unfortunately one of the highest in the developing world, and that pose a major challenge for the medicinal chemist to come forward with new medicine. At the same time, nutritionists and healthcare professionals to make sure that these diseases are abated through public awareness. Sadly so, drug discovery and development has always been, especially during the time of pharmaceutical industry revolution, become a business. So major investments are made in diseases which are common in developed countries, so-called Richmond diseases, and very little research and development expenditures are made in areas which are unfortunately called poor man diseases or neglected diseases. As a result, you'll find a whole range of different diseases which with no proper cure. Malaria is a classical example in which we have not been able to develop vaccines so far, though malaria kills more people than any pandemic every year. Leishmaniasis and other diseases are also very, very common, and for that there is very little investment available. And that is where I really want to end my remarks by saying that this is the good enough reason for us to develop our own indigenous capacity for medicinal chemistry and for our own capacity to develop drugs of our own diseases. This meeting is one step towards that. It is a capacity building event in which some of the most distinguished speakers in the field of medicinal chemistry from 3 continents would be speaking about uh, their work in the field of medicinal chemistry along with the strategies they would be following. So this is more often a workshop and at the same time a seminar. We intend to have more of these meetings. We plan to have a workshop. Originally, this was planned as a workshop, but when with the Delta variant, as a 4th wave of pandemic has emerged, we decided to change the mode, keep it completely online and convert that from a workshop to an online meeting because of the obvious reason. Workshop requires more of an interactive discussion, and that is always good with the hybrid mode. So with this, I would like to thank my very hardworking, committed, intelligent colleagues, Fazima Muazzam from Comstech and Dr. Hina Siddiqui from the International Center for Chemical and Biological Sciences for putting together an excellent program. And at the same time, I would also like to thank these wonderful speakers who spare time in very odd hours of the day for them. Some of them are really in the night zone and some of them early in the morning, but they're all here for the common cause of developing understanding, linkages, networking, and capacity of the people who wish to learn. And this is the joy of learning. And we all together will be able to make some difference. And I'm sure this event would be an exciting event. I would also like to thank participants for waking up so early. I'm sure that many of them would join us because Comstech World is spread from South America to South Asia, Southeast Asia, and that is a time zone in which we have to work. I'm sure many more of them will be joining us. So thank you very much, and I wish you all the best for this work, for this meeting. Assalamu alaikum wa rahmatullahi wa barakatuh.
Thank you very much, sir, for your insightful and comprehensive address. With this, I would request our technical focal person, Dr. Hina Siddiqui from ICCBS, for her vote of thanks and to introduce her in a brief. She's the brain behind this online meeting, and she is associate professor at ICCBS. Over to you, Dr. Hina.
Assalamu alaikum everyone, good day. Honorable Coordinator General, my dear teacher, sir, eminent speakers, faculty members, students, ladies and gentlemen. It's my privilege to have been asked to deliver a vote of thanks in this online meeting on recent development in drug medicinal chemistry, which is jointly organized by Comstech and ICCBS. I would like to convey my gratitude to the President of Pakistan, who is the chairman of Comstech, for his continuous support to the Comstech. It is my and Director ICCBS and my very dear teacher, Sir Professor Dr. Mohammad Iqbal Chaudhry, for his constant support, guidance, and encouragement to organize this event. Thank you so much, sir. I'm very pleased to extend our sincere thanks to the entire team of Comstech, especially Syed Aftab Hussain Zaidi Sahib, who is a Director HR and Admin, for his support in organization of this event. A very big thanks goes to Ms. Fazima Mozem, Program Manager Comstech, for her tireless efforts and coordination towards the organization of this very important meeting. Thank you, Fazima. I must mention sense of appreciation to all the resource persons who are very eminent scientists for their kind acceptance to be part of this event despite difficult time zones. Thank you, everyone. I'll be failed in my duty if I would not thank enough to all my IT support Especially, I would like to convey my thanks to Mr. Shamshad, Mr. Usman, Mr. Sahir. Thank you, guys. In the end, I would like to thank all the participants for joining in. I do hope to have you all till the end of this 2 days session. And I wish you very exciting, informative, and stimulating event in following 2 days. Thank you so much.
Very much, Dr. Henna, for your tireless efforts, as well as for for very generous appreciation. With this, we will move forward with our very first, with the beginning of our technical session. And for the first, we have Professor Dr. Parank with us. And with this, I would request Professor Dr. Iqbal Chaudhary to kindly introduce our guest speaker for today, so we can start our technical session.
Good afternoon, good evening, Professor Kekavas Prang, a dear friend from California, USA. He has been a great source of strength for Comstech, has been a great supporter of activities of Comstech because he has this universality and global vision of helping young people all around. I've had the honor of visiting Professor Brang laboratory, and I found scholars from all different continents, and that actually represent his motivation to help people all around. His presence here in very odd hours for him is reflective of his vision and his commitment. Dr. Brang is a scholar par excellence. He has been an active contributor of not only very high quality science, but also at the same time developing institutions and academic programs. And here we find his expertise very relevant because Comstech is in the process of not only establishing capacity through events like this, but also to seek expertise of people of his caliber for institutional review, looking into the syllabi, designing programs, and helping young people to engage in productive activities through joint cooperation. Professor Brang is Associate Dean of Research, Innovation, and Global Affairs and a full professor of medicinal chemistry and pharmacology at Chapman University School of Pharmacy, Irvine, California. Chapman is a private institution of a great vision. It is an institution with a clear understanding and a universal character. He earned his pharmacy degree from Tehran University of Medical Sciences, and I wish to remind you that Tehran University is one of the oldest universities in West Asia, and also a very prestigious one in 1989. He received his PhD in medicinal chemistry from the Faculty of Pharmacy at the University of Alberta in 1997, followed by a postdoctoral study in the field of solid phase organic synthesis in the Department of Chemistry, University of Alberta. He pursued additional postdoctoral studies at the Rockefeller University in New York and Johns Hopkins University in Baltimore. He joined the University of Rhode Island in October 2000 and became a full professor in July 2008. He served as a program coordinator of Rhode Island IDEA Network of Biomedical Research Excellence, which was a major NIH initiative which he looked after for several years. He joined Chapman University to assist in establishment of the School of Pharmacy. He is an author of over 200 research papers, 10 patents, 170 meeting abstracts. So he has been a very prolific writer of very high quality publication. Specific research areas currently under investigation include using peptides as cell-penetrating molecular transporters in the drug delivery, designing protein kinase inhibitors, developing multifunctional antiviral, anti-cancer, antibacterial agent. I remember my discussion with him, and he was mentioning about Professor Ehrlich's early dream of developing a magic bullet, as a molecule which is capable of treating many diseases. So he's actually on that very path. Uh, he also worked on designing a peptide nanomaterial for nanomedicine, one major area currently under investigation to design peptide nanomaterial for application in drug delivery. So we looked into the profile of Dr. Prang, and we found that with his very strong background in pharmacy, organic chemistry, and pharmacology, he is one ideal medicinal chemist which is equipped with tools and knowledge and understanding of various subjects required for this interdisciplinary science. He has been instrumental in developing lots of collaboration, initially of University of Rhode Island and now in Chapman University. And I'm sure that you would enjoy his talk. He has been a very inspiring speaker. So Dr. Parang, over to you, sir.
Thank you so much, Dr. Iqbal. Can you hear me?
Yes.
Yeah, okay. Thank you so much for the invitation and also for great introduction. And I want to thank Dr. Siddiqui and Kazime for organizing and supporting this event. And I hope I can give you some information that by the end of my talk, you can use it in your research and also your education. So thank you so much. I just want to share my screen. Can you see my screen?
Yes.
Okay. So the title of my talk is about prodrugs, and I'm sure many of you are familiar with this. So as Dr. Chaudhary mentioned, I'm working at Chapman University. Chapman University is in Southern California. And also I had another talk a few weeks ago. I'd be delighted to host any of you who come to California anytime and network and talk about collaboration in future. So let's talk about prodrugs. As you know, prodrugs are inactive compounds which are converted to active compound in the body. And this concept has been around for a long time. So usually we have a drug that is the active moiety and we mask it with the carrier or pro-moiety that can be converted back to drugs. Sometimes the conversion is done by chemical process, and sometimes we have enzymatic trigger or other type of trigger. So we're going to talk about that shortly. So in general, when you have a drug, and as you know, there are many small molecule drugs have been developed, and many of them have a significant biological activity, but we always have a challenge with absorption, distribution, metabolism, elimination, and toxicity that we call it ADMET. And many of drugs in clinical trial fail, even they are very potent, because of many of issue we have with each one of these. There could be limited absorption, extensive metabolism, or fast elimination, or toxicity, and lack of tolerance in the patient, and so on. So the prodrug is actually a smart concept that we can use to modify admit property and without compromising the drug biological activity. And this is a general way to improve the biological activity of the compound that have desirable efficacy, but not desirable admit property. So if we want to show that in general, we have an active drug and we have a barrier. This barrier, it could be a phospholipid bilayer barrier. It could be different other type of barrier. It could be blood-brain barrier, and also we may have poor permeability of the drugs across the cell membrane. So we have to find a way to bypass this different type of barrier to make sure the drug can have good absorption. And some of these barriers I mentioned here, we have limited solubility of the compound, we have poor permeability, we have also toxicity barrier, we have poor physical chemical property of compound, and sometimes we have metabolism and extracellular metabolism of the compound. So the compound is already metabolized before it reaches to the target. And sometimes we want to have a controlled release property, a sustained release property, and so we can have a longer acting effect of the compound. Or we might want to have direct targeting, for example, in many anti-cancer compounds that have extensive toxicity in other tissue. So the concept is very simple. As you know, we have a drug that attach it to promoiety and make it as an inactive compound. We call it prodrug. This prodrug has a good physical chemical property to bypass many of these barriers that I mentioned and be delivered to the cells, and then undergo different type of biotransformation or chemical transformation and generate the active drug and carrier. The most important thing that we need to consider, this promoiety or carrier has to be inactive. It shouldn't have toxicity, and also it should be biodegradable. So we don't have, we may solve a problem, but we don't rise another problem by generating a toxic chemical moiety. So this promoiety has to be eliminated, and active drug can be delivered to the body, bypassing many of these barriers that I mentioned. So the prodrug concept is not novel. And indeed, we had the first prodrug in 1853. As you know, aspirin is very old drug, acetylsalicylic acid, and it was marketed in 1899. At that time, we didn't know it's a prodrug, but eventually we found out that actually it's converted to salicylic acid that, as we know, it causes GI irritation, and aspirin has less toxicity in GI tract. But if we go back even to 1899, we had a compound methenamine that commercialized. That is actually, we're talking about 120 years ago. That is amazing that they came up with this concept that methenamine can be used for urinary tract infection. So they make this compound intentionally in 1899. And in the urine, we have acidic environment, and it can generate formaldehyde, and this formaldehyde can have anti-infective activity against bacteria. So again, 1899, we have the first intentional prodrug, but as acetylsalicylic acid was around 50 years earlier than that. But then we have 1931, 25, we have this antibiotic prantosil. As you know, it was discovered and by accident for the sulfonamide derivative antibacterial. In 1958, actually, the term prodrug was introduced by Adrian Albert. So even we had prodrug before, but nobody used this term before. And this was the first time the person who talked about prodrug as an inactive moiety that can be converted to active drug. And after that, we have an explosion of many different type of prodrugs came to the market, and many of them become some of the best-selling drug in the market, as you know. And this continue until today, that I'm going to give you several examples shortly. So in general, all of you know that the prodrugs have three components. One component is drug with functional group. The second component, you have a carrier or promoiety. And these two are connected with each other with a linker. And this cleavable linker can be degraded in vivo in the body and generate active drug inside the body. So we need to have these three components, and sometimes we call this is a classical form of prodrug. Sometimes we don't have a linker. Sometimes the prodrug is not actually generated by degradation. Sometimes the prodrug is generated by anabolism. So the compound we have that actually itself is a prodrug and has to undergo metabolism to generate the active drug. So this illustration is mostly a classical way to show it, but later on I'm going to show you different type of prodrugs that they don't follow this concept. And this kind of prodrugs, they are actually, we call them bio precursor. So they actually converted to active drug when they go into the body later on. So they don't have a link, they have to undergo metabolism. Okay, so if we want to look at the role of prodrug in the pharmaceutical industry, about 10% of all the drugs actually are prodrugs. Many of them are small molecule drugs, and this is increasing every year. So we have so many prodrugs on the market that you may not realize that we've been using them, but then they act as an inactive compound and they will convert to the active drug in the body. So there are many classes of the compound. I just gave some example here: antibacterial, antiviral, anti-cancer, many cardiovascular disease drugs that are used against cardiovascular diseases. So these are some examples of antibacterial prodrug, pivapicillin, becampicillin, hetacillin, and these are, as you can see here, these are ester prodrugs. So they are a derivative of ampicillin, and they've been using them to improve the bioavailability of many antibiotics or cephalosporin derivative. But sometimes we have a prodrug like this compound, saltamicillin, that actually is a combination of two different drugs. Or sometimes, as I mentioned, we have some simple drug like isoniazid that it doesn't have a linker, but it will be converted in the body to active drug after metabolism. The same thing, this is isoniazid, as you know, is used against tuberculosis And we have metronidazole that active against Helicobacter. And this compound is also not active by itself. It has to undergo metabolism to be converted to antibacterial agent. This is an antibiotic, ceftaroline, that this compound actually has a phosphate group. And again, I'm going to talk about this shortly. So this is kind of a compound that is used to improve the physical chemical property and improve the delivery of the antibiotic. So there are many antibacterial prodrugs. These are just some examples. Similarly, we have a lot of antiviral prodrugs. Antiviral prodrugs have been used for a long time. We have ganciclovir, panciclovir, and many anti-HIV agents like zidovudine was the first anti-HIV agent was developed. But now we have emtricitabine, lamivudine, and zalcitabine. Again, these are kind of prodrugs that I mentioned earlier. They don't have a linker and they are not attached to any carrier. They themselves are a prodrug, but when they get into the infections cells like HIV infected cells, they have to be activated, for example, for anti-HIV agent, or for herpes, they have to be activated. And this type of anti-HIV drugs or antiviral drugs, these are nucleoside derivatives, they have to undergo phosphorylation, and they get into the DNA of the cells and the viruses, and they cause chain termination. So the phosphorylation happens and they become active. So they are not a classical type of prodrug that they have a link. So if we look at the common functional group that we have for prodrugs, you can see that many of these compounds, they have a functional group like alcohol, carboxylic acid, amine, carbonyl, or thiol derivative. And then we have different type of linker that can be attached to this functional group and generate the carbonate, ester, amide, carbamide, and so on, even disulfide. And the commonly used functional group and linker usually are carboxylic acid and alcohol to generate ester derivative. But then we also have a lot of hydrazone and imine and thioether and disulfide bridge. So this, as I told you, these are in the drugs, and this is the linker. And then we have some release trigger that can be used to separate the active drug from the linker. So several of them could be chemical hydrolysis and pH, but also we have several conditions in our body that can be manipulated to deliver the drug selectively to desirable target. So this could be enzymatic. For example, many cancer cells have a specific overexpressed enzyme that can be used to deliver cancer anti-cancer drug to them. Could be pH. So we have an endosome inside the cell that can release a linker from the active drug. We have reduction and oxidation species and reactive oxygen species that they can be involved in release of many of these functional groups from the linker. So these are also different type of trigger that we can use. So if you want to have a smart delivery to a specific target, we can use different type of trigger to make sure the drug is actually released in the target, not in a place that is not desirable and cause toxicity. So we're going to talk later on a little bit about this shortly. So why we using prodrugs? There are many advantages of using prodrugs that we can categorize in 5 different groups: pharmacokinetic property, formulation property, also optimization, pharmacodynamic, and toxicity. Pharmacokinetic property, as I told you, we're trying to improve the ADME property, absorption, elimination, metabolism, improving the uptake, and so on, passing the blood-brain barrier. Formulation property, sometimes you have limited solubility, sometimes you have some functional group that undergo metabolism so quickly, so we want to mask them. Or they are very polar, they don't pass the membrane or blood-brain barrier, so we want to make them more hydrophobic. Or on the other side, we can also improve the water solubility. Sometimes we want to improve the water solubility of the compound because the compound are very hydrophobic and they cannot be delivered. Also, there are a process optimization. Sometimes there are some drugs that are in the market, and as Dr. Chaudhary mentioned, there is a major concept to repurpose many of these drugs rather than to go to develop new type of drugs. So you can use a commercially available drug and improve the physical, chemical, or biological activity and generate new drugs. But then because many of these prodrugs undergo conversion to active drug, the regulatory process will be much simplified because we already know the safety profile and toxicity of that drug. Many pharmaceutical companies, they also use prodrugs to extend the patent coverage. So they modify the structure a little bit and they can generate some new chemical entity and develop it with some improvement in biological activity and physical chemical property as a new drug and extend the marketability of active drug they already have. There are pharmacodynamic application. As I told you, you want to improve metabolism. You want to also allow intracellular conversion. So this is more about pharmacology and improving the biological activity and also reducing the toxicity of the compound. And as I told you, we can use the trigger-based release and smart delivery to reduce the toxicity. So with that, let me give you some example of highlighted application of prodrugs. We can improve the membrane permeability, we can prolong activity, reduce the toxicity, improve the water solubility or reduce the water solubility, and drug targeting and improving chemical stability. So I'm going to give you some example of this, and after this, I'm going to talk about a little bit about the different type of prodrugs that are in the market and also some emerging prodrug that develop based on the new technology, especially nanomedicine and enzyme-mediated release. And after that, I'm going to talk a little bit about my research that related to prodrugs, just a few applications that we have used in our research. So the first group of prodrug that application is improved membrane permeability. I'm sure you are aware that we have a drug called levodopa that is used against Parkinson's disease. And as you know, the dopamine is useful in treating a Parkinson's disease, but it's too polar to cross the cell membrane and blood-brain barrier. So if you use dopamine directly to the patient, you don't get very high concentration in the brain cell for treatment of Parkinson's disease. So what they did, they used the L-DOPA. L-DOPA has very similar structure with dopamine, except you have a carboxylic acid here. And this carboxylic acid is very sensitive to decarboxylation with the DOPA decarboxylase. So if you use L-DOPA or levodopa, it's going to be decarboxylated, and then it's going to generate dopamine. But the problem is that we want to deliver L-DOPA inside the brain cell first. So they use a carbidopa. Carbidopa actually inhibit the decarboxylate, DOPA decarboxylase, and L-DOPA can get to the brain cells and then undergo metabolism over there and generate dopamine. And then that can be used for treatment of the Parkinson. So L-DOPA is actually a prodrug to improve the membrane permeability, but it's always used with the carbidopa to inhibit the decarboxylate. The way it works is there is a transporter for L-DOPA. So this L-DOPA can be bound to the transporter protein and pass the blood-brain barrier and be transported and then undergo metabolism and generate the dopamine inside the nerve cell. So that is a specific transporter that can be used for this prodrug to be delivered to the nerve cell. So another example that I'm giving here is for improving the membrane permeability. A drug called enalapril. This is an angiotensin converting enzyme inhibitor that is used for high blood pressure and hypertension. So this compound has a ethyl ester group, and the active drug is a carboxylic acid. The active drug has a very low oral bioavailability, but when you add the ester group, you have an improved bioavailability by 60%. And then also you can use it as a tablet. But the active drug, enalapril, it can be used only by IV because it has a very short half-life. So just with simple modification, this can be converted to active drug in the presence of esterase, and this carboxylic acid is required for bonding to angiotensin converting enzyme. So that we have an active moiety functional group that cause the efficacy of this compound. So this is another example to improve the membrane permeability. Sometimes we use prodrug to prolong the activity. And one example is 6-mercaptopurine is a compound that has a thiol group. It's a very old drug. It's immunosuppressive agent, but it has a very short half-life and eliminated from the body quickly. So what they did, they developed this imidazole, 4-nitroimidazole derivative, attach it to the thiol group, and it shows a slow conversion to 6-mercaptopurine, and it has a longer half-life. And the person who developed this type of prodrug won Nobel Prize in 1988. And this compound, again, it has commercially available as a tablet, so you can put oral bioavailability and a slow conversion to active drug. And this undergo further metabolism converted to 6-thioguanine, that this compound later on was found to have anti-cancer activity against leukemia. So it generated a new area of anti-cancer research later on. So the third application that I'm going to talk about is using prodrugs to mask toxicity and side effects. So one simple example, as I mentioned, is aspirin. It's a prodrug of acetyl salicylic acid that is converted to salicylic acid. As you know, salicylic acid is anti-inflammatory and but it causes stomach ulcers and irritation in GI tract because of this phenolic acidic group, weak acidic group. And on the other hand, when you mask it with the acetyl group, it's hydrolyzed by esterase in the bloodstream slowly. So that is one of the most commonly used drugs that we have in the market. So another example that you may also have heard is cyclophosphamide. Cyclophosphamide is an anti-cancer agent. The area of alkylating agent in cancer, when you have nitrogen mastered, that your nitrogen that is attached to two chlorine group with the two methylene group. These compounds are very active and they have very extensive toxicity. So they came up with this idea that we make this nitrogen mastered less active. And then this has undergo metabolism in the liver. And in the liver, it slowly, several steps, it generates the nitrogen master that is, has alkylating activity, but much more selective compared to the alkylating agent. Many of alkylating agents that are in the market, they are used by injectable form because they are very active. If you use them orally, they cause significant GI toxicity. So cyclophosphamide is one of the alkylating agent that is actually non-toxic prodrug and is used as oral administration also. And it undergo metabolism with the liver with cytochrome P450 and is converted to active master derivative. So it reduces GI toxicity and it can be used orally compared to other DNA alkylating agents that cause alkylation of the different guanine bases in DNA. So this is a product actually that has been developed to reduce the toxicity of alkylating agent. Okay, so the next application that we have is a product to lower water solubility of the compound. And one simple example that you all know, chloramphenicol palmitate is just a prodrug of chloramphenicol that is converted by pancreatic lipase to active drug. And this is used for improvement of the taste, but we want to reduce the water solubility. Because when it's water soluble, it has a very nasty taste, and they make it as a suspension, so reduce the solubility in the saliva and is less soluble on the tongue, and you don't feel the taste of this compound when you consume it, especially for children that are very sensitive to bad taste of the drug. So this is to lower water solubility. On the other hand, we can improve also water solubility of many prodrugs. Another different example of chloramphenicol, we have chloramphenicol succinate. Chloramphenicol succinate, the succinate is added to chloramphenicol to improve the solubility because you get a sodium salt, sodium carboxylate salt, succinate salt of chloramphenicol that undergo hydrolysis with esterase and generate chloramphenicol, and sodium succinate that is not toxic. So this is because we want to improve the water solubility for IV injection, and also we can inject higher concentration, a smaller dose, and because it's more water soluble, is not a hydrophobic and lipid form, we have a reduced pain at site of injection. So this is another example of antibiotic that has been used as a prodrug for improving the water solubility. The next application also, I can give you example, clindamycin phosphate is another antibiotic. Again, we have a phosphate group here. This phosphate group is acidic, and in the physiological pH, it becomes ionized, 7.4, and we have negatively charged phosphate, and it become more water soluble. So clindamycin become more available for drug absorption. So again, this is another example of antibacterial product. And also because of water solubility, if it is used as an injectable form, it's less painful on injection. Another application of prodrug is to target different type of drugs that, as I mentioned, for example, at the beginning, we have metonymin, and hexamine is the same name. This compound, one of the oldest prodrug that was intentionally developed. And this is a very interesting molecule that when it goes to the acidic environment, it gets hydrolyzed, but is stable and inactive at pH 7 more than 5. So if you consume this, as far as the pH is more than 5, the compound is stable. But when it goes to the urinary tract, pH is less than 5, and it degrades and generates a formaldehyde, and formaldehyde has antibacterial agent. An interesting thing that formaldehyde is very toxic compound, that if you consume methanol that converted to formaldehyde, it can cause blindness. But this compound is so selective that it doesn't generate formaldehyde in the blood, and it goes to the urinary tract to generate formaldehyde. So the last group that I want to talk about is prodrug to increase chemical stability of the compound. And this compound is heterocillin, is another antibiotic, and this is converted to ampicillin. Ampicillin has interesting biological activity, but the problem is that this nitrogen, that amino group can attack to this beta-lactam and open this ring and make the ampicillin inactive. So they came up with this idea, maybe we should block this amino group and put it part of the lock nitrogen in heterocillin. So this is not active, or it's kind of rigid. It's become a secondary amino group rather than primary amino group, so it cannot affect this beta-lactam. And as you know, this ampicillin, beta-lactam are important antibiotics that inhibit the cell wall synthesis, and this beta-lactam is important. So this is a prodrug to increase the chemical stability of ampicillin. So ampicillin itself is chemically unstable because this nitrogen group can attach to the beta-lactam ring. Okay, so nitrogen atom again in this heterocillin is locked up and within the heterocyclic, and we reduce this activity. Okay, so this is the first part of my talk. So by this time, you should know what are different applications of prodrugs. And I only gave you examples about 7 of them, but again, there are many, many different applications for prodrugs. Is there any question at this time? Or I can continue again. If you have any question, I can answer now, or otherwise I continue. Okay, look like there is no question. please don't hesitate to ask me any question anytime, and I'm sure
Professor,
Fatima will looking
I'm
at
sorry
the
to
chat
interrupt.
and to see whether there is any question. Okay, but now you know the application of proteroids and you know how important are proteroids in different biological activity and physical chemical improvement and so on. But we need to know what type of proteroid we want to develop. There are different types of proteroids. One type of proteroid are carrier link or promoter. That is a classical one that I mentioned. And another one are bioprecursor that are different type of proteroids that we're going to talk about it shortly. For the carrier link or promoter, we have different type of class of proteroid. We have bipartite proteroid, we have tripartite proteroid, and we have mutual proteroid or double proteroids, or we call them also codroids. So each one of these have different application. So what I'm going to do, I'm going to talk about the carrier link type of proteroid and then go to the bioprecursor proteroids and tell you some examples of it. So carrier link proteroids are just classical proteroids that I already mentioned to you. So you have a drug with a functional group attached with the promoter, and you can attach them directly, or you can have a linker between them. If they are attached directly, we call them bipartite. If you have additional promoter, we call them tripartite. And both of these inert link carrier or promoter will be degraded and undergo hydrolysis or enzymatic or chemical transformation and generate drug and promoter. In this case, you have three sections. You have first promoter and second promoter. The second promoter undergo enzymatic separation, and then you have a chemical hydrolysis spontaneously generate the drug. So we have enzymatic and chemical together. Okay, so it's just a two-step process. So we're going to talk about these two first and give you some examples. So one typical example about the bipartite proteroid we can call famciclovir. Famciclovir has a 2-acetyl group, okay, and this active drug is directly attached to the promoter, and esterase is going to cleave this acetyl group and generate the active compound that undergo metabolism and oxidase form a pancyclovir. This pancyclovir itself is also proteroid. It's converted to monophosphate, diphosphate, and triphosphate that is DNA polymerase inhibitor. But the original proteroid is actually a bipartite proteroid that is converted to pancyclovir by hydrolysis of acetyl group and then oxidation. So there is no linker between the active moiety and this acetyl group. This is available as a tablet in commercially available. Another interesting example is ceftaroline is an antibiotic that has a phosphate group, and it can pass the membrane, plasma membrane, and then this phosphate group is going to be dephosphorylated by phosphatase and converted to active antibiotic that is ceftaroline. And again, this is just active moiety connected to the drug moiety directly. There is no linker between them. So that's why this is a bipartite proteroid. Tripartite prodrugs, as I mentioned, you have a drug, you have a carrier and linker. Again, is inactive. At the first step, you have enzymatic process, removing the carrier, and then you have a spontaneous chemical hydrolysis and generate the drug. So the first step is a chemical process. The second step is spontaneous chemical degradation. So I give you some example of this shortly, but this is commonly used, especially when you have an enzyme that is present in a specific tissue or organ, and you want, for example, this compound to get hydrolyzed only in that tissue. So you can make it very selective for targeting. So one good example of this is the ampicillin. Ampicillin is, again, another antibiotic that has a drug linking structure and carrier all together. So the way it works, this is ampicillin derivative, but it has an ester group and then another ester group, another diester group here. So you have chemical enzymatic hydrolysis of this ester group. So you by esterase, and you have conversion of this compound released of ethyl group to ethanol. And then you have this negative charge that undergo a spontaneous reaction and generate ampicillin immediately. So you have enzymatic and then a spontaneous chemical intermolecular chemical reaction that generate the active antibody. Again, you have a drug, you have a linking structure, and you have another carrier that is get hydrolyzed by enzyme. Okay. So now you know what is bipartite and what is tripartite prodrug. Then we have mutual prodrugs, another kind of carrier prodrug. In this case, we have two pharmacological active agents attached to each other. So you have one parent drug that has its own biological activity, has its own metabolism. You have another drug have biological activity, it has its own metabolism. And then when you have in vivo, they are separated and you have one parent drug and another drug. Both of them could have biological activity at the same time. So we call this mutual prodrug. The thing is that these two parent and drug could be attached to each other directly. So they could be bipartite, or they could have a linker between them, so they could be tripartite. But the reason we call them mutual prodrug because we have two drugs, or some people, they call it double prodrug. Some people, they call them co-drugs. So one good example is esramustin is an anti-cancer agent against prostate cancer. So you have estradiol attached to the nitrogen mustard at the same time. And when it undergo hydrolysis, you have a mustard and estradiol. And again, the compound used to for metastatic carcinoma prostate, and again, you use estradiol actually to deliver this to the prostate because it's a steroid. And also, this compound itself has some slow effect on the prostate cell growth, so it can reduce the growth of the prostate cell. So you have two kind of anti-cancer agent at the same time. Another good example is combination of cephalosporin and ciprofloxacin. This is a mutual prodrug that as you notice, you have ciprofloxacin here and you have cephalosporin, and this beta-lactamase can open the beta-lactam group, and then we have this electron delocalization that actually kick out the ciprofloxacin. So we have a beta-lactamase mediated activation, enzymatic activation, and you generate ciprofloxacin and also cephalotin analog. Both of them have antibiotic activity. So this way you have a double prodrug at the same time. Again, this is kind of mutual prodrug. So there are other examples of the mutual prodrug you can find commercially available. One is benoriolate. Benoriolate is actually anti-inflammatory agent. So you have a attachment of aspirin and acetaminophen at the same time through the ester group. So you generate a synergistic effect from two anti-inflammatory agents. In this case, sultamicillin, that is interesting compound. That this compound actually, sultamicillin, this is has a sulbactam and also ampicillin. The sulbactam is attached to ampicillin, but this is beta-lactamase inhibitor. So it inhibits the beta-lactamase for resistant bacteria, specifically that inactivate the ampicillin. So it reduces inactivation of ampicillin at the same time. So you have a compound, a prodrug, that one drug actually inhibits the metabolism of the other part. So the compound can remain active and go to the cell wall of the bacteria and can have antibacterial activity. So that's and the first group of prodrug that is carrier prodrug that was bipartite, tripartite, and mutual prodrug. But as you remember, I mentioned that we have different type of prodrug. We call them bioprecursor. These are actually innate molecule. They undergo chemical modification, but they don't have a carrier. Usually they become activated by metabolism, like reduction, oxidation, or activated by different enzymes. A classical example of this is Prantosil. You remember Alexander Domag, who was working in Bayer company, and they were looking for some antibiotic, and this guy used this compound for infection for his daughter. His daughter got infected, and he used this without informing the company. And then he realized that it has antibiotic activity. Because of generation of sulfonamide, Prantosil is actually metabolized to this sulfonylamide and also generate aniline derivative. So this generated a new generation of sulfonamide derivative that we already use them even now. Another example I just mentioned a few minutes ago, cyclophosphamide, that is activated in liver and generate the master derivative, and it generates the oral bioavailability of the alkylating agent. Again, we don't have any carrier here. The compound is undergo metabolism and become active. Cyclovir is another example of bioprecursor, and many antiviral drugs fall into this category. So acyclovir, as you know, is used against the herpes simplex virus. There is an enzyme, a specific enzyme, thymidine kinase in the viruses. So it phosphorylates acyclovir, and then you have a diphosphorylation and triphosphorylation that cause chain termination in DNA. The major interesting enzyme is a thymidine kinase that is in viruses. So only viruses can exclusively phosphorylate this compound and make it active. The other cells that are not infected, they are not going to do this. So that's why acyclovir, even is an old drug, is still used as antiviral drug because it's quite selective for the herpes that has a thymidine kinase. If you have a resistant herpes that you have lack of thymidine kinase, then acyclovir cannot work. So we have to use other different type of antiviral drug. But again, this is a bioprecursor for drug because it's not active by itself, is activated by phosphorylation, diphosphorylation, and triphosphorylation. So far what I have talked to you is about the classical example of prodrugs. And there are a new development in the area of the prodrugs that I want you to have some idea because many of these emerging technologies are in clinical trial or they're coming to the market shortly. They are a little bit more complex compared to small molecule type of prodrug, but they have significantly more extensive application. So you may have heard about prodrug-based self-assembly nanomaterial or enzyme-specific functional prodrugs. So these are two classes that I'm going to talk to you in brief, so you have some knowledge about this when this come to literature, and also you can use them in your research. So in general, when we're talking about conjugating a drug to a carrier amphiphilic polymer or other type of nanoparticle that these polymers can undergo self-assembly. So this self-assembly, as you know, there is a very important process. So when we're talking about self-assembly, consider that cell membrane is a self-assembled structure of phospholipid bilayer. DNA is a self-assembled double-stranded chain that we have halogen bonding between the different nucleotide bases. So the same concept here, we can use amphiphilic compound to self-assemble, and we use this for specific application. In general, for nanomedicine, we can reduce the renal clearance because you have a larger molecule. These larger molecules have less tendency to metabolize fast and undergo filtration in the kidney. Also, we reduce the metabolism because we protect the drug. So the enzymatic metabolism will be diminished or reduced because the drug is encapsulated or self-assembled inside the package. One major application is you can use multiple or tunable drug release mechanism. So that way you can have a specific delivery to tissue, and you can smartly adjust it. So you can make the drug release based on the condition you have. It could be externally or it could be internally. So you can have an external factor to make the drug release, or you can have internal factors that are available in tissue environment to make the drug release. And this can happen using the nanomaterial. And again, there are many different nanomaterial. You can do surface modification and make them more selective to target For example, you can have a targeting ligand on the surface of nanomaterial that can go only to a specific tissue. Traditionally, we have used antibody to target different type of tissue selectively, but now we know that antibody are not exclusively selective and they have some their own issue, and cost of synthesis of those compound antibody drug conjugate is much more compared to if using a prodrug concept. Okay, so let me give you some example of various type of nanoparticle drug delivery system. So you have polymeric nanomaterial, we have lipid-based nanomaterial, you have also inorganic nanocarrier. And many of these have been developed or have been used previously. For example, liposome, solid lipid nanoparticle, micelles, and so on have been around for a long time. And each one of them have major advantage and disadvantage in terms of leakage, in terms of how much loading they can provide for the encapsulation and so on. More application coming through for polymer. Polymers are more diverse. You can have different type of polymer. So when I'm talking about polymer, it's not only synthetic compound. It could be also peptide. It could be different type of polymers that can be self-assembled, and hydrogel, dendrimer, and so on, and they can be used for encapsulating of drugs. These are two different classes of nanoparticle drug delivery system. Inorganic nanocarrier has been also used, but these are mostly used for imaging, but some of them, like gold nanoparticles, are very good biodegradable and not very toxic. And some of them, like magnetic nanoparticle, has been used for delivery and also tunable delivery of the compound. So if you can use the heat and you can use the magnetic nanoparticle, there are thermosensitive stimulus release of compound. Carbon nanotube was a hot topic a few years ago, but then we know that single wall or multi-wall carbon nanotube, there was a controversial about the toxicity of this compound. Quantum dot has been used mostly for imaging application, but there are mostly inorganic cadmium that are usually toxic, but they have a very good imaging property. So how about we use this application of nanomedicine with the application of prodrug at the same time and come up with some new type of delivery system. So we call them prodrug-based nanomedicine. And they can have a stimuli responsive release of the compound. So you can have a prodrug based on the polymer. You can have prodrug based on the small molecule, or you can have an encapsulation of nanomedicine. So we call this emerging area prodrug-based nanomedicine. And this is becoming a very diverse application for not only delivery, but also for targeting specifically to tissue using the stimuli responsive pathway. So I give you some example how this is working. So if you're talking about different type of prodrug nanoparticle, you can do it two different ways. So you can have a prodrug itself that is a classical prodrug that we have and encapsulate it inside the nanocarrier. Encapsulation of this prodrug inside nanocarrier, you can have a prodrug encapsulated nanoparticle. This is one way to do it. The second way to do it, we don't need a nanocarrier. The drug, the prodrug itself, it can self-assemble by itself. and generate a nanoparticle by itself. So we have this compound that it could be attached to amphiphilic compound, polymer, peptide, and so on, and self-assemble and generate nanoparticles. So when we're talking about self-assemble of compounds that are amphiphilic, we're talking about amphiphilic that have charge or hydrophobic residue. So hydrophobic residue love each other, and charge residue, they can have hydrogen bonding with each other, and we have a nanoparticle like this. Okay. So let's give you one example of this doxorubicin conjugate that undergoes self-assembly. So you have a oligoethylene glycol polymer here, and you have a linker, and you have a doxorubicin that is attached to the amino group. So you have an imine group here. So this hydrophilic group, they are on the surface. They want to be exposed to the water molecule, and this doxorubicin that is blocked amino group is inside, and we have this linker. So when this undergoes self-assembly, you have this nanoparticle. But then when it goes to the endosome, that is acidic property, this imine group will be cleaved, and this amino group will be released, become polar, and will be kicked out of the endosome because of positive charge. So this way you can deliver doxorubicin, even it goes to the endosome, but because acidic environment of endosome and lysosome, it can be released. So this nanoparticle is formed for delivery, but when it goes to the cell, it will going to degrade it, and doxorubicin that is in the middle will be released. So that is kind of combination of prodrugs and nanomedicine that improve the delivery at the same time. The second class of the emerging technology are enzyme-specific functional prodrugs. So in this way, we have several drugs, especially this is used on oncology. We have anti-cancer drugs that are very cytotoxic. We have doxorubicin, paclitaxel, and so on. Then we have a cleavable linker. This cleavable linker is sensitive to a specific enzyme. So these are not just a chemical linker that what we had in traditional prodrugs. There are very specific amino acid or glycosidic bond or so on that are sensitive to a specific enzyme. And then you have additional targeting moiety. So you can have targeting ligand or light responsive moiety or other nanoparticle at the same time. So these are enzyme specific because they only act in the presence of a specific enzyme to cleave this and release the drug. They are quite stable unless we have exposure to a specific enzyme. Let's give you one example here. Again, I have here doxorubicin. And as you know, doxorubicin is a very commonly used anti-cancer drug, and it has a red color. And you can actually see the red color when it's injected under the skin. And that's why even when you make a nanoparticle, you can actually see the red color in nanoparticle. So you see here it's attached to a linker that is 4 amino acid, phenylalanine, arginine, and glycine. We call it FRRG. So this linker is very sensitive to cathepsin B. Cathepsin B is a cysteine protease that only cleaves a specific sequence of amino acid. So this conjugate doxorubicin and the linker, it has a property that can self-assemble with each other. So you have doxorubicin in the middle and this linker also outside So you have a nanoparticle, but then in the presence of cathepsin, this is degraded. And cathepsin is present in many cancer cells. Many cancer cells have overexpressed cathepsin B. So they can cleave this nanoparticle much better compared to the normal cell. So you generate some kind of selectivity here. Okay. So right now it's 11:07. So I gave you some background about the classical prodrugs and some imaging technology. The imaging technology is a little bit more complex because you have multiple components. So in terms of pharmaceutical industry, they may be more challenging to develop this compound, but there are many of them in clinical trial. But the advantage is that you can have more selectivity, more protection in presence of different metabolizing enzyme and better delivery. So in the remaining minute, I think I have to finish for the next talk and give you some time for question. Give you quick example of prodrug in my research. So recently we published a paper in kind of similar to some of this imaging technology, that we have a peptide that has a positively charged arginine on the head and 2 long chain fatty acid. And this peptide generates a kind of nanoparticle that we can use it to encapsulate sRNA. So sRNA, and you look at electron microscopy, you can see the cavity in the middle, and then you have a bilayer outside. So this sRNA, we can actually compare. If you have free sRNA, we don't have much delivery to the cell. If you have lipofectamine, that is a transfecting agent that is commonly used. If you have a delivery of sRNA to the cell, but as you know, lipofectamine is kind of toxic. You cannot use it in the body. And then you have our peptide lipid nanoparticle. We had 2 different types of them, and we will be able to deliver sRNA to the cell. But this type of plan A, we call it plan A, they are non-toxic compared to lipofectamine. So you can have fluorescence labeled sRNA inside the cell. And if we want to see the silencing of the protein, we have compared this. So if you have a protein, no treatment, lipofectamine can be used to silence the production of the protein. This sRNA bound to mRNA and degraded. And in presence of our delivery system, also we had silencing. If we compare to the lipofectamine, it's comparable to lipofectamine. We have tested in also other protein like SARC that we can silence SARC protein also similar to lipofectamine. So if it's no treatment and in the presence of this delivery system. So the second application of prodrug that I want to talk about is nucleoside analog that anti-HIV agent. So we have fatty acid that have some modest anti-HIV activity that we discovered. And then they actually inhibit an enzyme in the HIV that involved in methylation, so of different protein in the HIV. So this fatty acid can be conjugated to this type of compound. So we have methylation of the protein inhibited by fatty acid, and then we have anti-HIV nucleoside that inhibit reverse transcriptase. And we realized when we conjugate this together, we can inhibit 2 different enzymes, N-methyl transferase and reverse transcriptase. So we have done synthesis of many fatty acid conjugate of different type of nucleoside. And if we look at the parent drug, lamivudine, and then fatty acid conjugated you see that the activity is significantly improved. If the acylation is in the hydroxy group, you have significant improvement compared to the parent drug against both X4 and R5 type of viruses. So you have a lamivudine that IC50 is around 7.5, 2.6, and then with a fatty acid conjugate is 0.3, 0.082. Again, you have emtricitabine, it's an anti-HIV drug, and we have a fatty acid conjugate that improves the activity at least 20-fold more. And if this kind of compound also active against the clinical isolate, so we can see that the conjugate is significantly potent compared to the fatty acid plus lamivudine in physical mixture. Again, the conjugate is more potent than physical mixture. This is also active against multi-drug resistant HIV viruses. This is lamivudine, and this is a conjugate. So as you can see, we have kind of a double prodrugs here that fatty acid conjugate actually improve the delivery of nucleoside to the HIV infected cell. But on the other hand, the fatty acid itself has also some biological activity. So that's why we get significant improvement compared to the parent analog. Animal study has been done by a different group. They look at the drug level in biopsy mice, infected mice. And then if you have the parent drug release in the presence of the fatty acid conjugate, you see from 0 to 4 days, we still have release of lamivudine. But then the parent drug has very limited release. If you go 4 to 14 days, we still have release, but then the parent drug almost 0. And you can see the production of lamivudine in different tissue like liver, spleen, and lymphatic tissue. That is many places like lymphatic tissue that is actually the hidden reservoir for HIV that can be penetrated by this type of product. Another final example I'm going to give you is a doxorubicin, is an anti-cancer drug. This compound bound to the DNA and also topoisomerase and intercalate to the DNA and inhibit the topoisomerase. And this compound has some major problem. One is cardiotoxicity. And we were thinking that how we can improve the delivery. And also the second problem is resistance. Many cancer cells also like the doxorubicin. So a few weeks ago, I gave a talk and I just here summarized. So we have a peptide that conjugate to doxorubicin. This is actually a prodrug that is attached to a linker. So we have a tripartite prodrug. And this compound actually generates doxorubicin inside the cell. And if you go for 24 hours incubation with ovarian cancer, we don't see much doxorubicin alone, but then conjugate, we still have a lot of doxorubicin released inside the cancer cell. We have higher retention, so we reduce the efflux. And the activity is very similar to doxorubicin. If you have a conjugate, you get improved activity at longer period of time, but then it gets released. So you have a conjugate slowly converted to doxorubicin until completely converted to doxorubicin 99%. That's a doxorubicin standard. This is a conjugate standard. We incubate the cell to this conjugate. At the beginning, we have a conjugate, very small amount doxorubicin. But again, you will see the reduction of the conjugate and release of doxorubicin until it's converted completely to doxorubicin. Another conjugate that we have a hybrid cyclic linear peptide attached to doxorubicin. It has a very good uptake to ovarian cancer cell, but again, it has not much uptake to heart cell, cardiac cell. That's surprisingly, but that's a good advantage because doxorubicin itself has a very high uptake to heart cell and cause cardiotoxicity. So as I mentioned earlier, we can use the prodrug to reduce the toxicity of compound, and this tripartite prodrug is one good example that we use in our lab. Another example, we have conjugate doxorubicin with a large peptide WR9 through a linker, and this peptide actually have itself protein tyrosine kinase inhibitory activity, so it's anti-cancer activity. So if we use the doxorubicin resistant cell, doxorubicin resistant cells, doxorubicin has no activity against this cell, not much. But when you have the conjugate, you see even at 1 macromolar, you can see after 72 hours significant activity. Here, doxorubicin even after 72 hours has no activity. So you can use this approach to bypass the resistant mechanism by doxorubicin. So as you can see, doxorubicin doesn't get to this cell line because it's resistant. But then if you have doxorubicin conjugate, as I said, it's red, you can see it inside the cell. Even if you mix doxorubicin with this peptide as a physical mixture, you can see that delivery inside the cell. So it can be used for the delivery of doxorubicin at the same time. Okay, so with that, I just want to conclude. We have several types of products have reached to the market. And as I showed you, there are multiple applications that you saw here in different ways. Some of them are classical and some of them are emerging technology. And these applications, some of them are to improve the pharmacokinetic and pharmacodynamic drawbacks of active drug. The new technology like nanomedicine has been merged with the prodrug right now and generating novel therapy to be used for difficult to treat indication. So this is a very active area of research, and many of you may already be working on the prodrug application without being aware of that this is a application that have been used. Recently, we have a collaboration with Dr. Siddiqui and some of the PhD students that we are working on the developing prodrugs of anti-HIV agent in Pakistan. So there is a lot of application we should think about, and I hope this talk give you some motivation and think about your area of research, that how you can combine it with the prodrug application. So thank you so much. So I'd be happy to answer any question, but I think we have a few minutes. But again, I want to thank the organizer for inviting me. And if you have any question, you can email me anytime. Thank you so much.
Thank you very much, Professor Dr. Parang. It was such a wonderful and comprehensive presentation. Hello? Professor Parang, can you hear me?
Hello.
Hello, Dr. Heno.
Yeah, can hear me?
Okay.
Yeah.
Oh, okay. Thank you very much, sir, for your wonderful presentation. It was really detailed and comprehensive, and I think very well to understand actually. So we have some questions here. Let me read them out for you. First of all, we have question from Ms. Noor. She was asking that, okay, so first question is, sir, can a mutual prodrug be referred to as a chimeric molecule or a hybrid molecule? Hello?
Yeah, you can use hybrid molecule, when we're talking about hybrid molecule, we're talking about conjugation of two different molecules. But again, if you look at different type of prodrug, if you're talking about bipartite or tripartite, these are hybrid, or mature prodrug, these are hybrid molecules. But then if you're thinking about bioprecursor, that is a different type of prodrug, they are not always hybrid. They have their own entity. They have to become activated inside the body to become active. So not all the time there are hybrid molecules. So if you're talking about bioprecursor, there are a different type of products.
Right. So here is another question.
Yes.
What is the fate of pro-moiety once it has delivered the drug?
Okay, that's a good question also. As I mentioned earlier, we need to be very careful when we have a pro-moiety that is released. It has to be non-toxic and it has to be biodegradable. Usually many of these pro-moiety are metabolized and eliminated from the body. For example, I gave you example of fatty acid or ethyl ester or very simple pro-moiety that are biodegradable. And they can be eliminated. So the fate of this compound, if they are, if we are not seeking activity in pro-moiety, they have to be biodegradable and metabolized and eliminated.
Thank you. So there is another question from Mr. Noor Rahman. He's asking, is there any advantages of prodrug over the nanocarriers?
Advantage of prodrug over nanocarrier, yeah, there is advantage. Usually nanocarrier application is a little bit more complex, and you have to engineer and manipulate the release of the compound, and you have to have a specific size of nanoparticle. And usually simple classical prodrugs are much easier to synthesize. In terms of pharmaceutical industry, manufacturing is much easier. But if you're talking about nanoparticle, even we have several nanoparticles already in the market, you know, to convince the regulatory agency to have a standard for this nanoparticle and make sure they don't have toxicity based on the nanoparticle is a little bit more complicated because it's an emerging technology. But right now, in terms of pharmaceutical manufacturing, prodrugs are much easier to manufacture, and regulatory agencies are easier to approve them because they already know the activity of the active drug. But when you go into the more complex nanoparticle, you have to prove also that your nanoparticle is not toxic and it releases the compound specifically in the way you want it.
Okay, thank you. Here is another question from Mr. Azhar Abbas. He's asking, kindly explain why are you calling inactive compounds also as a prodrug? I think he sort of wants to ask what is the difference between inactive compound and prodrugs.
Okay, so inactive compound, when we're talking about inactive compound, we have two different inactive compounds. So inactive compound that they don't have any activity and they can be eliminated. They don't get converted to anything. They just metabolize and leave the body. But when we're talking about prodrug, they are inactive drugs. They have the capacity to be converted to active compound. So there is a significant difference for something that intrinsically inactive and it never gets active compared to something like a prodrug that is inactive, but it can be converted to active compound. So there is a distinct difference, a thin line between them. But why we call it prodrug inactive? because they will be converted to active compound, but we don't call them inactive compound, we call them inactive drug.
Right. So here is another question from Mr. Kishore. He's asking what will be the best method to deliver atropine-like drug through nanotechnology?
Deliver what type of drugs?
Atropine, A-T-R-O-P-I-N-E, atropine, or atropine.
Atropine drug. So this we're talking about anticholinergic drug. I don't recall the structure specifically right now, but I can tell you in general, if they are hydrophobic drug, so you can think about a nanoparticle that have a hydrophobic core and a hydrophobic shell, hydrophilic shell. But if there are you're talking about a compound that is hydrophilic and you want to deliver it with nanoparticles, so you want to design a nanoparticle that has hydrophilic core or hydrophobic shell. So you have to look at the physical chemical property of your drug and make your nanoparticle based on that. But at this moment, I don't recall the structure of atropine, but I remember it's a very simple structure. Look at the functional group of the compound, look at the lipophilicity and polar functional group, and see what type of nanoparticle you want to. Why do you want to use a nanoparticle? Are you planning to improve the delivery, or are you planning to improve the physical chemical property? Are you planning to improve the circulation time? And based on that, design your nanoparticle based on that. Yeah.
Right. So he has another question. What would be best polymer for the ophthalmic delivery of polar drugs?
Polar drug, if you're talking about polar drug that you want to make sure that they have a better circulation time, so I will suggest to use some polymer that have a hydrophilic core and hydrophobic shell. Example, most of the polar drug, for example, if you're talking about peptide, they are biodegradable, or some polar drug that are biodegradable. Many people use polyethylene glycol to cap the drugs, so it completely protects the drug from the hydrolysis. It's also water soluble, so it doesn't generate precipitation. So one example, I can use polyethylene glycol, but there are other polymers like PLGA, polylactic acid, also they have been used, and they are very good biodegradable that can be used. But again, it all depends on type of drug, because even polar drugs have different physical chemical property, and they may undergo different type of metabolism. But you can think about some hydrophilic polyethylene glycol or PLGA.
Thank you very much, sir. With this, we're sort of running short in time, and we are very grateful for your time, and it was such a wonderful talk. And with this, I would request Professor Dr. Iqbal Chaudhry for his final comments. And again, thanking you for your valuable time.
Thank you so much.
Dear Professor Parang, thank you very much for yet another mesmerizing presentation with lots of knowledge. It was indeed more of an excellent understanding which people have developed through the introduction and then strengthened with the examples which we have provided. So I'm sure this is a lecture which people would like to play again and again to learn more and more. So thank you very much.
Yeah, thank you, Dr. Iqbal.
I'm so extremely grateful. You can have a good sleep now because it's already late in California. Thank you very much, Professor Dr. Kaikubas Frank.
Yeah, have a great day. Yeah, thank you.
Thank you, sir. Thank you.
Thank you very much. Thank you, sir. With this, I would request our next speaker, Professor Dr. Khalid M. Khan, for his lecture. The title of his topic today is Medicinal Chemistry for New Alpha Glucosidase and Alpha Amylase Inhibitors. And with this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, to introduce our guest for our speaker today, Professor Dr. Khalid.
Bismillah ar-Rahman ar-Rahim. Khalid is a dear colleague. We have spent almost half of our life together. He has been associated with me since 80s, and I am always very impressed from his extremely hardworking nature and his capacity to do things in a very nice manner. Khalid is among the most prominent medicinal chemists of the country. He has done wonderful work and trained very large number of individuals in Pakistan. Along with this, he has been engaged with me in the promotion of chemistry as a discipline. So I am associated with Khalid, and it's such a pleasure. Khalid is a professor of organic chemistry. He's among the most prominent researchers of Pakistan, made seminal contribution in the field of medicinal chemistry and drug discovery. He received many prestigious awards and honors, including Civil Award, Sitare-e-Imtiaz and Tamga-e-Imtiaz. He has been among the most cited scientists of the country, and he has served as the president of, as the Secretary General of Chemical Society of Pakistan. He has been associated with National Core Group in Science and Technology, which was a major initiative for the promotion of science and technology, chemistry as a discipline and its application in industry. He's mentoring a very large number of young researchers in the field of drug discovery and development in a very interdisciplinary manner, was successful in establishing large number of productive collaboration. He has trained over 90 PhD, MS and MPhil scholars. So he has, to his credit, a whole generation of people who got trained from him. Khalid was able to introduce several novel classes of pharmacophore in the world literature. And during this process, he used a minimum pharmacophore approach, which is an approach in which you rely on the use of minimum chemicals. At the same time, you build and structure, which is in terms of carbon value, is more drug-like. He has rationally designed a number of new lead molecules, which has been active against diabetes and peptic ulcer. He has published lots of experimental studies in top international journals. Khalid is active in synthetic organic chemistry since long, and I consider him as a mentor for large number of young people. His credit include 800 research publications in Impact Factor Journal. He has been cited on a very large scale, about 17,000 citations with an impact factor of 1545. So certainly he has been most prolific scientist and a most accomplished one. Along with this, he has also patented 18 IPRs in different countries of the world. Khalid would be speaking to us on a very interesting topic related to his recent work. Professor Dr. Khalid. Khalid, you are mute.
Yes, thank you, Professor Iqbal Chaudhry. I'm very grateful to you to invite me for this wonderful event, which I am looking more than 200 people are participating at the moment. So, and I am also thankful to Khazima and Hina because they are continuously in contact with me, and they are asking about what are your these things and that. So I would like to say I would like to share. I would like to share my presentation with you, and I will talk on the medicinal chemistry of new alpha glucosidase and alpha amylase inhibitors. Thank you very much, Professor Bajodi, once again. And I'm very much excited to say that Professor Bajodi, because we always say to you, Dr. Saab, and I am saying you, Professor Bajodi, because it is a meeting, so I have to say that. So where is the share? Very open है, open है, very. Okay, so we have to share. Where is the meeting? Meeting come. Yes. Okay. Medicinal chemistry of new alpha glucosidase and alpha amylase inhibitor. I will talk to you people something about the lead molecules. Lead molecules are lead compounds. Lead compound is a chemical compound that has pharmacological or biological activities. It can be used as an initial point for the chemical alteration in order to increase potency and selectivity. Lead is a substance which is not a drug, but is used in medicine, biotechnology, and pharmacology in the course of drug discovery. So my topic is alpha amylase and alpha glucosidase because both are involved in the diabetic mellitus. Diabetic mellitus is the most prevalent metabolic syndrome worldwide with an incidence varying between 1 to 8%. Diabetes is characterized by the hyperglycemia resulting in various short-term metabolic changes in lipid and protein metabolism and long-term irreversible vascular changes. Diabetic mellitus, there are two types, type 1 versus type 2. Type 1 diabetes occurs when pancreas is unable to produce enough insulin. That means the pancreas is not producing the insulin which is required for the body to digest or to the carbohydrates tend to develop at a young age, cannot be prevented, require insulin therapy. Type 2 diabetes occur due to insulin resistance when the body does not respond well to insulin. Tend to develop at an older age, but nowadays it is not the case. Sometimes this develop earlier, can be prevented with lifestyle changes, can be managed with lifestyle modification alone if diagnosed early. Both share symptoms of frequent urination, increased thirst, extreme hunger, unintentional weight loss, fatigue, blurry vision, sores or wounds that heal slowly, and numbness and tingling sensation in hand and feet. Both can benefit from lifestyle modifications such as healthy diet, physical activity, blood sugar level monitoring, and management of stress and other existing health conditions. Diabetes can develop many disorders like affect eyes, cataracts, blood vessel nerves, neuropathy, and kidney nephropathy, and cause impaired wound healing. As I told earlier, life expectancy is only 2/3. In Pakistan, 20% population of over 30 years are of age has impaired glucose tolerance. Other disorders with the diabetes mellitus are beta cell damage, increased blood sugar, increased rate of production of AGEs and cross-linking, heart diseases, endothelium fibrogens, and collagen fiber are destroyed. Alzheimer's disease, amyloid proteins are side products of Amadori Schiff and Maillard reaction. Cancer, acrylamide, and other side products are released. Peripheral neuropathy, the myelin is attacked, and the deafness, demyelination, and blindness, microvascular damages in the retina. Current strategies for the management of diabetes: reducing insulin resistance, increasing endogenous insulin production, exogenous insulin supplies, limiting the postprandial glucose absorption. Management of diabetes. Diabetes can be controlled through enzymatic or non-enzymatic pathways. There are two ways. Enzymatic pathway involve inhibition of enzymes that are hydrolyzing the carbohydrate into glucose. For example, amylase, alpha-glucosidase, beta-glucosidase, and non-enzymatic pathway involve anti-glycating agent that prevent the formation of AGEs, advanced glycation in end products. There are some medications are available in the market: sulfonylurea, metformin, thiazolidine, dione, acarbose, maglitol, and etc. World Health Organization announced that in 2003 versus 2005, in 2003, 194 million people had the diabetes, but by 2025, 333 million are expected to have it. But during the years and last 2.5 decades, we are targeting different molecules for different enzyme inhibitions. The main is alpha-glucosidase, and the second is the alpha-amylase. And our target is like this, that easily accessible starting material, because we are living in a third world country and where we are very much difficulties to getting the starting material for the synthesis. Therefore, we thought that we have to have a very easily accessible starting material to work with. One of two, we don't want to go in a long step synthesis, so we always prefer one or two step synthesis of the simple molecule, very simple molecule. And for the purification, only we try to get the compound like that may be crystallized easily or distillation. Cost effective, easily, and if these all things have a compound, so then that will be easily adaptable for the pharma industry. Enzyme inhibition, key tool for drug development for a wide range of enzyme-related diseases. More than 30% of drugs in clinical use are enzyme inhibitors. Many pesticides and insecticides also work as enzyme inhibitors in the target organism. Plant and other living sources can provide novel and potent enzyme inhibitors. And we always use the ELISA reader to work with, and using the measurement of UV for the measurement absorption at 400 nanometer. Enzyme inhibition is studied concerned with the enzyme substrate reaction influenced by the existence of any organic or biosynthetic compound or an inorganic metal caused by their covalent or non-covalent interaction with active site of enzyme is called enzyme inhibition. Compounds that control the rate of enzyme catalyzed reactions are called modulators, moderators, or modifiers. The effect of this control is to reduce the rate, and this is called the inhibition. Enzyme inhibitors are used as pharmaceutical agents in human and veterinary medicine, as well as herbicides and pesticides. Many antibiotics are also enzyme inhibitors. The inhibition of an enzyme involves a molecule or drug which reduce or block its catalysis. Basically, we want to block the catalysis of the enzyme because the enzymes are biocatalysts. There are four main classes of enzyme inhibition: competitive inhibition, non-competitive inhibition, uncompetitive inhibition, suicidal inhibition. Competitive means inhibitors compete with the substrate for binding to the active site of enzyme. Inhibitor binds to the free enzyme, not to the enzyme substrate complex. This is the overview for this, that enzyme substrate come and make a substrate bind with active site of enzyme molecule, and then reaction occur and produce molecule are generated. But in inhibition, inhibitor come into the log, then and it remain with that and prevent the binding of substrate molecule. Non-competitive inhibitor is a molecule that binds to an enzyme anywhere other than the active site. This alters the enzyme's three-dimensional structure in such a way that its active site can bind to the substrate with unusual affinity, but not able to stabilize transition state and catalyze the reaction. In this way, substrate enters active site of enzyme, but active site and this make this unusual binding. That's why the substrate cannot produce the or catalyze the hydrolysis. Inhibitor only bind uncompetitive, only bind to enzyme substrate complex for enzyme inhibition. In fact, inhibitor does not affect binding of substrate directly, means it attacks somewhere else, anywhere, and can disturb the 3D complex of the substrate and enzyme. And suicidal inhibition is mechanism-based inhibition and is form of a specific irreversible enzyme inhibition. It follows an enzyme bind a substrate and forms an irreversible complex through a covalent bond during the catalysis. Aspirin, which inhibits cycloogenase 1 and 2 enzymes, penicillin, which inhibit DD transpeptide from the building bacterial cell wall, and sulbactam, which prohibit penicillin resistance, strain of bacteria from the metabolizing function. So these are 4 types of enzyme inhibition. How alpha glucosidase inhibitors can help? Alpha glucosidase is present in the brush border membrane of the small intestine. It catalyzes the final step of carbohydrate digestion so that its inhibition suppresses the release of glucose from the dietary origin. The catalytic role of alpha glucosidase makes it therapeutic target to treat carbohydrate mediated diseases. Inhibition of alpha glucosidase delays the rate of conversion of disaccharide. That means it delays the conversion of disaccharide into monosaccharide. As a result, the postprandial mean after meal blood sugar glucose level is maintained at a lower level, leading to decreased insulin demand. It means that it delayed the digestion process, which means the absorption of glucose would not take place into the blood, and the glucose level will not increase so much, and it will not need the more insulin to decrease the blood sugar level. This approach is useful to manage glycemic index independent to insulin in diabetic patients. Can be used as anti-obesity because the obesity always comes with the diabetics. So it also used for the anti-obesity drugs. Alpha glucosidase and alpha amylase inhibitors. Inhibition of intestinal enzymes that break down the carbohydrate does delay the absorption and digestion of carbohydrate, as I told you before, in the gut, especially target meal-related postprandial hyperglycemia and independent, which is an independent risk factor for the cardiovascular complication. Control the glucose level independently of insulin. The effect on glycated hemoglobin are comparable to metformin or thiazolidines. Alpha glucosidase inhibitors are initial treatment for patients with type 2 diabetes, cause no hyperglycemic events, cause no weight gain, potential to use as anti-obesity. Acarbose is one of the drugs which is marketed for this, and the second is maglitol. These are the structures of both drugs. And alpha amylase is a calcium metalloenzyme present in the saliva and pancreas of the human and some other mammals. Major digestive enzyme in animals catalyze the hydrolysis of long-chain carbohydrate, and the alpha amylase hydrolyze alpha-1,4 glycosidic linkage, thus yielding oligosaccharides as end product from various polysaccharides. New alpha glucosidase and alpha amylase inhibitor from our lab. This is the part that first was the introduction of alpha glucosidase and their complication, alpha amylase and complication due to their overexpressions. So and the next part is the new alpha glucosidase inhibitors and amylase inhibitors from our lab. So let's start from the acetylchalcones. This is a very interesting, these are the very interesting compounds, and we produce these compounds by the action of two carbonyl compounds, and we have got 29 compound of this, and we we published this in Biology, Medicinal Chemistry 2021. And this is the structure activity relationship between the compound, the carbos, the standard has the IC50 value 937 plus minus 1.6, and you could see the compound which we prepared having a pyridine ring have the 23.08 alpha amylase inhibition and 26.08 alpha glucosidase inhibition. And you can see the pyridine part is everywhere is the same, but the changes we are just modifying this terminal. The other terminal, which is, if we see the OME was present on a phenyl ring, there was this type of inhibition. And when we put 3 methoxy groups, so the inhibition slightly different, but there was enough inhibition of both enzymes. And if we introduce the fluorine at one of the phenyl positions, so we have seen that there is a very mild change, but all are the compounds which are active. And we introduce the nitro, so there is a slight change, but not very much decrease in the activity. And the case with the nitro plus chloro group is also the same. The second series which we have prepared is the synthesis of dihydroquinoline 4,1H on derivatives. This is very easy, one-step synthesis, and we got all 37 compounds in the solid form, and we recrystallized the compound with the help of from ethanol, and you can just see the various compounds. This is a 3-component reaction where we have just made these 1 to 3 37 compounds in a very efficient method, and we have published this in Molecular Diversity in 2021. And you can see the value of carbose is 937 as a constant, and we have seen we have the compound here. You can see, I once again go into this, then we changing this R. So we have to see the change in the activity like this here. Like this here, this is a chloro substituted compound where the chloro is on the ortho position of the compound. So it shows the activity 25.84 and 26. First is the glucosidase and second is the alpha amylase. 937, and you can see how much active are our compounds. And we change the compound, introduce one more chloro at the second ortho group, the activity remains about same. And when we introduce a compound, a chloro at the ortho para position, then slightly change in the activity, but the pattern remains same. And if we introduce the bromo at the para position, then we have the somewhat same activity as the compound 3 has. This is the and if we introduced the methyl O-methoxy group. So we have got some comparable activity there. But when we introduce the methyl and ethoxy groups at the position meta and para, the activity slightly decreases. This is another class of compound which is called the conazole analogs. So this we have prepared in one step, and using the triethylamine as a by catalyzing the triethylamine in ethanol 2 to 3 hours, and we have prepared about 21 compounds out of this, because this is a two-step reaction. First, we have prepared the conazole structure, and then we have derivatized the thio group of this with the help of many compounds, and we have published this in 2021. And this is the activity, but these are not those much active as we have previously, as I showed you previously. These are the only alpha glucosidase results, and this is 312 against 937, 377. Anyways, these all compounds are more active than the standard. So if we see some structure activity relationship, nitrogen containing phenyl with the O-ethyl group, it gives 312, and if it is with the chloro, it's 77, if CF3, 231, and if CF3 goes to the para position, it gives the 305, and if this is the CF3 is changed by the NO group, then it will be 191. And if the bromo is come on the meta position, it has the 85. And if the nitro group comes on the, and if we introduce this group, then it is 90 and etc. These are the all example of those. Synthesis of indole acetamide, this is one of the another class, and we have used this CDI for making for preparing the direct conversion of acid into the amide. And this we publish this work published separately, and for this matter we have used the same strategy and this methodology by using the CDI and converting the acid into the directly into the hydrozoids or amides. So in this particular slide, we want to tell you the indole acetamide can be formed and by this method, and 1 to 4 molecules have been prepared, and we have got these results. This amide is linked, N is linked with the parabromo compound, parabromophenyl. It has the 1.09, and if it has parafluoro, it has 1.09, and if it is a parachloro, it is 1.75. So these are the results which we have published in the ACS Omega in this year. This is another class of compound. This is the indole-based thiodiazoles, and these are the compounds. And we have published this year, and these are the results. These are alpha glucosidase results. 1.30 when this is fluoro is at the para position, and if it is at ortho position, it is 1.10. If it is nitro at the meta position, it is 1.60, and if it is dichloro, it is 0.95. So you can see the effectiveness of the different functional group on the activity of the molecules. And for your kind information, these are all non-cytotoxic compounds on different cell lines. This is of indole sulfonamide. This is very easy to prepare compound, and we have published this in bioorganic chemistry. You can see there is a amine, indole amine, and we just react those with the sulfonyl chlorides, and we got these compounds, these type of compounds, and you can see the activity of this compound. This, when it has the bromo at the ortho position and para is, and at para orthomethoxy and second meta methoxy, it is 1.60, and if it is ido and combination of ido and O-methoxy, it is 7.30, and if it is combination of chloro or nitro, it is 4.70. I'm just showing you those compounds which are most active. Benzimidazoles derivative. These are prepared in 3 steps. First, this is intermediate form, and this is the third one. Second one, and when we reacted both of the intermediate, then we formed this type of compound, and these are the activities. This is published in Journal of Molecular Structure 2021, and these are the results. When this is metal and nitro, and this is 1.40, 1.30, and when these are alpha glucosidase inhibition results. So these are the two fluoro group at para and ortho. It has the 0.90, and if these are the this ring is replaced, nitro group nitro ring is replaced with a other group, so then it is 1.60, and if it is nitro, then it is 1.60. This is another class of compound, indazole hydazides and sulfonahydazide. This is prepared in 2 steps, and we have published this in 2020, and you can see what is the structure activity relationship between them, and the modification on these points. And you can see when it is dimethyl, it is 1.99. If it's trifluoromethyl, it is 1.23. If it is ortho-nitro group, it is 1.66. and if it is paranitro, 1.37. And if it is combination of methyl and nitro, it is 1.93, and chloro at two ends, so it is 1.91 and 1.38. It is the position of the chloro is changed from the meta to para, so then it is 1.33. And if it is combination of dichloro and hydroxy, it is 1.252. And if it is alone methoxy group, it is 1.97. New indazole based derivative, this is another thing. And in this, we have reacted this compound with the thiocyanate, and we have did the modification of compound at the nitro group, and we found this type of compounds, and the activity relationship is this. If it is chloro at the meta position, and there are two types of inhibitions. One is alpha glucosidase and second is alpha amylase. This is, if it is at meta position, it is 1.62 for the alpha glucosidase and second is 1.71 respectively. Then if chloro is at para position, it is 5.2 and 1.67. There is no any change. If you can see actually there is no any change, but the activity is there, and we were preparing these compounds. Why? Because if one is cytotoxic, then we can use another one as a lead compound. So that's why we have modified many structures, just changing the position of the functional groups. 1.69, 1.82, bromo 1.8, 1.98, fluoro, the position is ortho 1.52, 1.62, and you can see there here is the fluoro and next fluoro. There is any very mild change in the activity. Dihydropyridones, this is a 3-component reaction, and we got this type of compound where this is the change position, the R group. You can see this R is changing everywhere. And this is the 2.58, the alpha glucosidase for this compound, where the para and meta methoxy groups are present, and where the two meta groups are present, 3.90 or 4.01. That means the change of position change the activity. So if this is a alpha benzyl group here, so ortho benzyl group here, so benzyloxy group, then 2.33 and 2.55. And if it's a combination of methoxy and fluoro group, then 3.02, 3. And you can see everywhere the groups are changing the position activity of the compounds. These are the amino 4-aryl phenyl thiopyrimidine 3,5-dicarbononitrile, and this was our project, and we have successfully completed this project. And this is published in 2020, and this is the very easy two-step synthesis, and we got these type of compounds and having the activity with the change of the groups. You can see these are the groups which we are going to change, and we are changing here, and here. And the most active compound was 103, I think, yes, 103, which has the nitro group at the at the meta position, and when it was methyl at the ortho position, it was changed to 273, and when the phenyl group changed by the pyridine group, it was 304. Because I am going slow, so I will skip some slides. Benzophenone 5L phenyl methanol derivative. This is one-step synthesis and published in the 2020. And these are the some examples of this compound, and you can see the alpha glucosidase activities 18.04, 19.46, 28.84, 29.86, 28.06, and this is just because of the changing of these functional groups: bromo, chloro, nitro, bromo, and chloro. The positions are changed. These are aryl oxidizole shift bases. This is three-step reaction, and we have published in 2029. And these are the some activities you can see: 0.30 versus 937, 0.6, 937, 1.1 versus 937, and 2.7. These are the most active compounds of the series. And this series was of, I think, 34 compounds, and we can see this, the bus changing the position of the group. You can see the difference. If it is at para position, a hydroxyl is at para position, you can see the 0.3 activity, and but when it is it goes to meta position, it is double of that, 0.3 and 0.6. And if it is nitro, it is 1.1, and the nitro at the alpha position, it goes worse. These are the other examples of this series, and I will not go into the detail. I am going to the next series, the 3-amino 1,3 oxidizole, and this is two-step synthesis, and we prepared these compounds, and you can see the alpha glucosidase. Alpha glucosidase activities of this compound, and I have to go fast because I have a lot of slides, but I have no time left. I am only 10 minutes left. So these are the different type of compounds, and I will just go indole, acrylonitrile, and other, and I think I have to just go to 2, 3, 4 slides, and then 1,3-dione. This is medicinal chemistry, and you can see the very excellent result from these. And I think I would like to give you some time for the question answer. So I will just touch two more series. This is benzamidazole derivative. This we have made by this like this, and these are some compounds with the alpha glucosidase activities, and you can see the 4.6, 19.1, just changing the hydroxyl group here, and 2.6 dihydroxyl group is a 9 to 5, 11.4, 25.4, 18.2. So barbiturate, this is a class of compound, and we produce this compound over here by modification on this position, and this you can see this is 41.95 against 935, 37.6 against 937, and this quinoline. This is also by medical chemistry paper in 29. I will just skip these slides and I will go benzohydrazide. One more is this thiazolyl coumarine, because the coumarine is the class which we have explored a lot of time, and we think that we have synthesized more than 1,000 compounds of coumarine. So therefore, I want to show this series of compounds, and you can see the compounds. This is the coumarine compound, coumarine containing compound, and you can see 1.10, 7.40, 2.90, 1.40, and 1.10, and lot of more. As diazole, these are those compounds from where we initially started in 1997, that the thiazole group was our group and our desired group at that time, and we prepared a lot of compound in this type of series, and you can see the compounds having the good activity of alpha glucosidase. Triazoindole, I will not go into the detail because I have left only 5 minutes, so I will just go to the last slide and give you the time for that. Concluding, we have successfully identified a range of lead molecules for the treatment of diabetes. Most of compounds are in their in vivo studies. 8 compounds are patented. And I am thankful to all of my collaborators, especially Professor Iqbal Chaudhry. I couldn't see the name of him, but I think that is upward, and Dr. Simon Rashid, Dr. Sanal Abbasi, Ms. Ambreen Khan, Mr. Hamdi Kastos, Dr. Ajmal Khan, synthesis of compound, Dr. Momin Khan, Dr. Fadlain, Dr. Zarbasha, Dr. Taha, Mr. Shahriyar Hamid, Mr. Irfan Ali, Mr. Shahbaz Shamim, Mr. Muhammad Ali, and I am thankful to financial support of Pakistan Academy of Sciences, Higher Education Commission, Pakistan Science Foundation, International Center for Chemical and Biological Sciences, Ministry of Science and Technology, and Sindh Higher Education Commission, International Organization of Prevention of Chemical Weapon, OPCW, the Netherlands, the World Academy of Sciences, International Science Foundation, DART, and many more. These are 2 people to whom I owe all the things which I achieved and got during my career. Thank you very much. And this is my small research group. Thank you very much. If you have any question, I am here. Stop sharing. Uzema, I am here.
Thank you very much, sir. It was a wonderful presentation, and thank you for your time. And again, you're doing such a wonderful work. And with this, the floor is open for question and answers. So we have some of the questions posted in the chat box as well. Let me read them out to you. The first question is, could you please tell how inhibition activity of glucosidase was evaluated?
How?
Inhibition activity of glucosidase was
first we will get the percentage inhibition, and then we will convert that by logging, by converting into calculating its IC50.
Okay. Thank you. We have another question. Kindly explain the docking significance in the new drug designing.
Yes, yeah, docking significance is very important because I didn't give any docking study into this because we did docking studies for all of our compounds, but this is very significant because you can predict the active interaction of your compound with the active side of compound by the help of computation in the studies.
Thank you very much, sir. It was a wonderful presentation. And as you can see, our participants are also posting in the chat box. They are really delighted and honored to hear you and your work. With this, I would request Professor Dr. Iqbal for his concluding remarks.
I am thankful to Dr. Iqbal once again for giving me this opportunity.
Thank you, sir. Please, sir.
Professor Dr. Khalid Khan, thank you very much. It's always a pleasure of listening to you. Thank you for sharing your very valuable experience in this important field. Zima, I would like to move forward.
Yes,
Khalid,
sir.
thank you.
Thank you very much, sir. Thank you, sir. With this, we will move on to our next speaker. Our next speaker is Professor Dr. Feng Chen. His title of the talk today is Metal-Based Anti-Tumor Agents of Traditional Chinese Medicine, Active Ingredients, and their derivatives. And with this, I would request Professor Dr. Iqbal to kindly introduce and welcome our guest speaker.
Dear colleague, I'm once again extremely delighted to introduce someone known to me since long, a very dear friend, someone who I cherish for his original contribution in the field of medicinal chemistry and organometallic chemistry, a friend and also a long-time collaborator, Professor Dr. Zhang Feng Chen, from a friendly university, a university with which we have a long-term collaboration. He is the director of the State Key Laboratory for Chemistry and Molecular Engineering of Medicinal Resources. So we work in various different disciplines of medicinal chemistry. It is part of the School of Chemistry and Pharmacy from Guangxi Normal University, Guilin in People's Republic of China. Professor Chen has visited Pakistan several times, and it was always a pleasure of welcoming him. He has received his PhD degree in organic chemistry from Nanjing University of China, which is a very prestigious institution. He has got this degree in 2001. Then he worked in Guangxi Normal University of China as an associate professor. And in 2003, he became a full professor with the research focus in medicinal chemistry. So his journey from PhD to full professor was rather short, and that shows his productivity and excellence. In 2004, he became the owner of program for New Century Excellent Talent in University of Chinese Ministry of Education. The second level honor of program for recipient of the program for New Century 10, 100, 1,000 distinguished talents. The Bagao Scholar of Guangxi Zhuang Autonomous Region in China. In 2012, he was approved as the leader of innovation team of Ministry of Education in China. And in 2015, he received a new program for National 100,000 and 10,000 Talent and National Outstanding Contribution in the Export of China. So these programs are for identification of the most talented young scholars in different disciplines and the person who is responsible of taking care of these programs are often very eminent scholar. In 2016, he was approved to receive the State Council Special Allowance. Currently, he is Executive Vice Dean of State Key Laboratory for Chemistry and Molecular Engineering of Medicinal Resources at Guangxi Normal University of China. I happen to be a Cheng professor there also. He's completed 4 NSF project of China, one in 973 project, and published more than 220 research papers in top peer-reviewed journals, 1 book chapter. He's also inventor of 53 authorized patents in China and abroad. Dr. Chen's major research interest is in drug discovery in the field of traditional Chinese medicine, and he has been focusing on cancer since long. He's trained people from all around the world. His laboratory is a common destination of scholars from Pakistan, Central Asia, and also from Africa. And it is always a pleasure of introducing Professor Chen, both because of his academic excellence and also for his personal friendship. Thank you very much, Professor Chen. Dear friend, Professor Chen, you are mute, I believe, because we can see your slides, but we cannot hear you.
Okay, thank you, Dr. Ikuba, give me a very long and very detailed and very kindly introduction. My talk title is Metal-based anti-tumor agents and of traditional Chinese medicine's active ingredients and their derivatives. And as you know, Pakistan, Karachi University and Guangxi Normal University, we have established a joint international laboratory. And during the several years, we have carried out a series cooperations in the PhD students coverage and project applications. Today, we will give about 10 parts reports on my recent research on the traditional Chinese medicine and metal-based anti-tumor agents research. And since from the 2019, well, the COVID-19 first discovered in the Wuhan, China, the virus, it is very tolerable and now significant influence our living and scientific research. Otherwise, here we will sit together with Ikoba and all colleagues to discuss our research. But now we have to stay here to talk about my research. And the virus, as you know, virus infect host cell must pass 6 steps. And the most important virus RNA and cell is DNA controls. So viral replication is very important. A virus cannot replicate on its own. It must be attached to an host cell. It then uses the host cell's energy to synthesize protein and DNA and RNA. And RNA viruses are typical kill because they live inside human cells. Any drug that kill a virus may also kill human cells. Actually, most important to worry about the viral infections. Actually, in our living, we must confront the viral infections. The component immunization based upon to the viral infections. A well-functioning immune system will emulate or effectively destroy virus replication. Immune compromise can help frequently viral infections. Cancer claims especially leukemia or lymphoma and transference due to the pharmacological therapy and so on. And there are some key characteristics of antiviral drugs able to enter the cell, infect with virus, interfere with viral nucleic acid synthesis and or regulation. Some agents inhibit the ability to viral to bind the cell. Some agents stimulate the body's immune system. And actually, bacterial virus infection and cancer have a very intensive relationship. Here we give some sample. Helicobacter pylori HP will induce carcinoma, and HPV will induce a hepatocellular carcinoma, and HSV will induce cervical cancer, and HPV will induce cervical cancer, and so on. So virus affect infections. It is very collectively with the cancer. And as you know, from the Chinese medicine to fight Chinese medicine to fight COVID-19, it is important in China. And the early period of COVID, our university, our government use Chinese medicine to kill the COVID-19 pandemic. And TCM is present in all the Chinese philosophical thinking. And they believe that the world is a single unit, and its movement of both yin and yang. Yin and yang are not absolute but relative. Drugs or herbs are used to carry the imbalance of yin and yang in the human body. And meanwhile, Western science and medicine are the recognizance that disease is analyzed at the cellular, molecular, and the pharmacological level. And as you know, Professor Tu Youyou comes from China, the owner of 2015 Nobel Prize in Physiology and Medicine. Why she obtained Nobel Prize? This because her discoveries concerning a natural therapy against the malaria, she obtained some inspiration from the Zhou Hou Beijifang cold and heat treatment of malaria by the Eastern Zheng Dynasty Guo Hong recorded. she from the artisanal coverage floor use ether to displace often to obtain the customer and firstly carried out the animal test. And up to now, treatments of platinum cancer and cancer drugs have been obtained. Since 1968, Luxembourg discovered the sixth patent has the very strong anti-cancer activity, and to 1978, USA FDA approved sixth patent in clinical applications, and 1989, USA FDA approved coupling, and in 1995, Japan approved NADA printing, and in 1998, China approved lob plating, and 2002, FDA approved oxyplating. Up to now, and Japan sun printing, China disc printing, and also up to now about more than 1,000 platinum compounds have been flown into the trial test, but up to now only 8 platinum and cancer drugs used in the clinical applications. And the major in the problems in the metal-based anti-cancer drugs and strategies. As you know, and many publications and problems is toxic and resistance. And in the our research area, we will focus on many strategies such as change DNA action mode, improve drug targeting, exploit medicinal potential of the metal, try the non-classical targets, and such as the protein, and so on, enzyme, and design to the material targets, combined with TCMs, and stratify and sex. We will use it to design the TCM metal-based and cancer agents. Here, we discuss some advantages of metal-based and cancer and TCM active ingredients. Platinum-based and cancer drugs have some advantages: unique DNA damage mechanism, coordination number and geometry diversity, various oxidation and reduction, thermodynamic and kinetic factors, metal ion and ligand factors. Also, they have some disadvantages: serious toxic and side effect resistance. On the other hand, TCM active ingredients, we call that TCMAI, as natural medicine play key role in cancer treatments. According to document reported from the 1930s to 2014, 175 kinds of small molecular anti-cancer drugs came to the market. 131 kinds of about 70% originate from the natural product and use the derivatives. And the TCM active ingredients contain some advantages: resource and species abundant, activity, structure diversity, multi-bioactive, and relative low toxicity. But they have some disadvantages: low content, delectability defect. So, and we also we focus on some new agents and the tumor targets DNA top 1 and top 2. Top 1, top base 1 and top 2, and the DNA also the platinum complex and some organic drugs such as the hydrocorpotency is the target to top 1 and top 2. And the G4 DNA as they are new and cancer targets. And the Nobel Prize in Biology and Medicine in the 29 was awarded by joint to the Elizabeth Blackburn, Carl Clay, and Jacques Jockett for the discovery of how chromosomes are protected by the telomerase and enzyme telomerase. And the currently research indicated that G4 DNA are highly associated with cancer, HIV, and diabetes. G4 DNA are involved in the regulation of telomerase activity that is activated in 90% and 80% to 90% of human tumors. So, and chemistry, the medicinal chemistry focus on the G4 DNA telomerase to design the new compound to examine their anti-cancer activity up to now. And and there are many of many organic compounds have been investigated for they are targeting the G4 DNA telomerase compared to the organic compounds. Metal complex G4 DNA in ligands have superior properties. Firstly, is the right style structure and favorable electron withdrawing metal unique photo ecology properties enhance water solubility. And however, the G4 DNA bind enter phase 2 clinical trial, but more in vivo data needed to make a more reliable prediction of their psychoactive relationship. And questions, there are questions, how about combine the modern medicine with TCMs? What inspiration from TCMs? And the TCM-AI plus active metal center. Targeting DNA, telomerase 1 and 2 G4 DNA, telomerase and other protein. We focus on this thinking. We carry out our research. The first part is the program metal and metal-based and tumor agents. And program and Jabra growth in 1 to obtain for treatment for rheumatoid arthritis and dystonia and boils and cancer. It has from this plan we is checked plant banking to synthesize the copper complex and by pleading a mixture ligand complex, and this compound can inhibit the top one activity in compared to program 2 complex is keep the remarkable enhanced inhibition of activity top 1 and at the 1.56 milliamp they can occur in populations. This publication is our group, the early publications on the TCM-AI metal-based and cancer agents. And then we focus on the lymphocyte metal complex. We synthesize a 5 pregnant complex, lymphocyte complex, and determine they are IC50 and intact and action mechanisms. And these compounds is keep the very good activity against the BCL 7404, and it's better than the 6 platons. And multi-metal-based and the tumor agents. Matri is 60 root of the sulfur catalyst gamma grows in Guangzhou of China. And the matri MT have been extensive use in China for treatment of various use in China for treatment viral hepatitis and cardiac disease. It keep the inhibition after many tumor cells. Tumor cells with iron and lead and copper and gold and garden complex. This complex almost in the ionic complex is 6 in the solid or solutions. And this compound keep the in vitro activity to the panel of cancer cells. And part 3, oxoapophyne, the metal-based agents. Oxoapophynes are a subclass of isochlorine chlorides and widely distributed in the medicinal plants of family and analysis and so on. Up to now, about 400 oxoapophyne has been discovered. Oxoapophyne have some structural characteristics: good planar modulator molecular protein, hydrophobic conjunctive signaling, nitrogen and carbonyl as well as substituted as group and parallel links. Oxoapophynes possess a broad range of biological activity, such as anti-cancer, anti-microbial, and viral and platelet aggregation inhibitors. And firstly, we extracted from the medicine plant Mangalore Guangxi, Zanfoxylin nitidant, usually liliaodan, the first compound that we extract from TCM. And we use liliaodan to alleviate on the platinum and the leucine, magnesium, iron, and zinc, and copper, and gold, and obtain a series of the metal complex and determine they are a critical structure, and then we investigate in vitro activity against the panel of cancer cells and determine they are action mechanism in cell level and molecular level. You can see this publication, and series publications. And here we focus on iron and magnesium, coal, zinc, and the complex able to inhibit the activity top 1, which times increase at 5 microgram top 1 logs the activity. And the long time gold complex induced BCL74704 and induced the S phase arrest. Lilodine, but the ligand lilodine induced a G1 phase arrest. And so the copper complex ionic gold and coordination gold is different. They are binding mode is different, and they are activity and the mechanism are different. and use the copper and this the rhodine co-ops with the platinum. Platinum 1 complex 1 is stabilized G4 DNA inhibit the active telomerase. And next natural compound is the oxochlorosin. Oxochlorosin is fixed in the paragraphs, and we use synthesis method to obtain this ligand to react with gold and a series transition metal to give the zinc, copper, and magnesium complex. And OG metal complex have a very low IC50, and but the ligand IC50 is larger than 150 microgram. It means OG has the very low activity to the cancer cells. And but the circulating, the IC50 is 25.3 μm. And from this sample, we can summarize that OG metal complex inhibit enhance the cytotoxicity. And oxochlorosin diasplein complex is the diasplein dy complex is effective inhibit tumor growth in BL7-405-02 is sinogram both inhibit higher safety in vivo than the cisplatin. And the third natural ligand is this combined we call LY is isochlorine chloride is 6 many kinds of TCMs with cancer activity. We use the 6 stability action to obtain to total synthesis this ligand. Use this ligand to react with Loden and obtain the Loden complex 2 induce the HAP2 in S phase, which includes CHK1 and SEC2, P21, P27, and P53 is present in decrease of cyclin A and CDK2. And induce a HAP-page apopts via caspase dependent mitochondrial pathway that solve is intrinsic pathway. And IC50 LY, the IC50 is 23 and 9 microgram, and but and complex 2 have has the 7.56 μm and increase threefold, but to the normal cells HL7702 very low toxicity. And the Loden complex effect inhibit tumor growth in HAP2 is underground mouse model with TGI 53.04%, longer than cyclin, but is exhibited higher safety in vivo than cisplatin. And the fourth natural ligand is also upon find called also upon find the latives. We synthesis also upon find the lative to obtain its copper zinc nucleic complex caused this complex caused the cancer cell death via cell cycle arrest in S phase, mitochondrial dysfunctions, and Among that, the nucleic compress is shipped allowed in weak bowel thoughts that then significant and a certain in weak bowel and cancer activity. And just now we just extracted or synthesis natural product, natural ligands. Here we focus on the parallel ligand and also upon find contain A, B, C, D tetraline. Tetraline we induce 3 amino 1,2 propanediol in position 4 obtained. They are 3 ligands SR ligands, and obtain the platinum compress 6 and 4 so high cytotoxicity to the BCL7O cells than the H normal cell. And this compress and induce BCL704 apoptosis in C micro G14 independent VTPH complex 6 induced telomerase damage and cell senescence and compensate a dose of 8 microgram kilogram and 5 flora have tumor inhibition rates of the 41 to a point% and 43.7%, but complex 6 inhibitor higher safe than the 5-fold. And we use the same before that, but change the metal center, from the platinum to leucine. We obtained and not the 3 leucine compress. This 3 compress have a very good water solubility and most selective for the BCL7-4 cells than the normal cell H normal cells. The selectivity as shown this table shown that the S configurations complex have a good better IC50. The level of complex on telomerase region is higher than that of complex 4 and complex 5, and complies with the order complex 6, 4, and 5, which also induce cell senescence. And FID and FET assays indicate that complex 4 to 6 is keep the highest selectivity for PU20 and HTG20G4 DNAs than for the other double DNAs. Complex 6 could inhibit the telomerase function, while downregulation the simulchromata, which was different from the complex 4 and complex 5. This is weak in the also complex have single behavior to BEL7 of a model with an inhibitor of later 53% lower than the secretant. But and this result public JMC. And summary 1,2 methyl plays key role in anti-cancer activity. Position 4 introduce chiral water soluble 3 amino 1,2 propanediol increase the solubility of complex in generate chiral configuration relationship. Position 19 introduce substitution group refractive complex activity. And another sample is oxo upon 5, oxo iso upon 5, oxo iso upon 5. Oxo iso upon 5 is 6 in the imperfect Dorican, we call DC. They skip the cancer activity. Also, iso upon 5 possess a broader range of biological activities such as cancer and bacterial immune function. Oxo upon 5 can become a new source of developing cancer and drugs. Also, look at the nitrogen, oxygen. Firstly, we synthesis the cobalt, the platinum complex. This very wonderful. You're looking PD1, PD2, very different, just a very less different. Here is hydrofox, and another is the hydrogen, but they are activity and mechanism is very different, and in vivo activity are different, and action mechanism are different. This sample, apparently told me that in the medicinal chemistry, we design the molecular with different substitution group. They are pharmacology activity are different, and the pharmacology mechanism are different. Okay, because another sample is the cobalt. We use the oxo upon 5 according to cobalt, and but increase the planet, increase the planet, we obtain copper 2. Copper 2 is keep the tumor growth, inhibit the infections, and the copper inhibit better safety profile than dispirtants. Also, due to the ligand planar increase, and copper 2 is keep the better binding activity to the G4 DNA. And now we focus on oxo isoapophyte site 11, site 10, site 8, site 6, and site 7. We synthesis a series oxoapophyte the lipase as a position 6, 8, 10, 11 substituent group change. What happened? We changed the metal center. What happened? We obtained, we synthesis and catalyzed 14 metal complex, platinum, nickel, and finally we obtained complex 2 and complex 3 and complex 6, and different from the position side, but the other samely, but they are activity are different, and the activity binding to the G4DNA and the in vivo and in vitro activity. Complex 2 is bigger complex 6, which also calls the mitochondria dysfunction in order 3. Complex 6, complex 3, bigger complex, large complex 6. And also complex compound, you have a very good inhibitor in vivo activity, but complex 3 inhibit the higher in vivo cell than complex 6. And we focus on 9% precision. 9. We introduce amino at 9 precision. We obtain the platinum, and this platinum complex inhibit very good G4 DNA and reproduction of mitochondrial functions. And we introduce the bromide, oxo isoapophyte obtain the platinum, also induce the inhibition of telomerase. summary. Also, isoapone by a position 4-5 fuse large plane will increase the binding activity to the G4 DNA. Also, isoapone by a position 6, 8, 9, 10, and 11 substituent groups. We will obtain different structure ligands and change the metal center. We obtain the copper, electron, nuclear paradigm, and so on with different buttons. What we will obtain, the different metal complex with different and cancer activity and action mechanism. Part 6 isoapone bond, we obtained the isoapone bond gold complex. This paper published by Pakistan PhD student Khan. And this London is Khan and Ecoba cooperated our research to obtain this Khan. Isoapone a chlorine paradon. This the kind of wife Nosi or obtained a PhD in our group. She obtained an isochlorine platinum compressor which good and cumulative was obtained through the microangial mediated prophecy and autophase death. Autophase and apoptosis diamotal death. The result published in CSS chemistry. CSS chemistry is the very important journal of China CSS associate chemistry, a journal. It is very difficult to publish. And we obtained to the organic mortality gold compressed with a similar skeletal structure. Golden iron replaced the first carbon atom in the nitrogen heterocycling ring of the THIQ obtained. Cycling cobra 2, gold 2 is kept low toxic, low listing factor sequence. And this results published in the JMC. And part 7, philosophy of philosophy and quinoline combines. We focus on this structure, and this structure in vivo response in IgD sowing. IgD in vitro and vivo flow interest. This results published in the chemical. And next is carbolene. Carbolene cobalt complex. Beta carbolines are widely distributed in the plants and marine animals. Beta carbolene are cross-molecularly derivatives and trickle consistent endophyllidine. Beta carbolene are cross-position and the activity, and we focus on beta active. Beta carbolene. Now, because the document says that MCO1 is an apoptotic member of BCL2 family of proteins. It plays an important role in the survival of a series of cancer cells. BCL2 inhibitor drug has been approved by the FDA with that classification by the FDA. And some small molecular focus on that targeting MCO1 have been reported, such as quinoline, sulfan derivatives. There are still no MCO inhibitor drug approved by FDA because the part of MCO1 is smaller than the BCL2 protein. Affinity receptor allowed, and no, except for our group, no metal-based MCO1 inhibitor has been reported. And as shown this protein structure so that hot spots of the MCL protein have P1, P2, P3, and argon 3 to argon 20, argon 263, and these crucial hotspots. How to attach MCL1? Actually, MCL1 inhibitor inhibits the hotspot to MCL1 protein. Inhibit the function of MCL1, activate the back and back dependent pathway, induce tumor cells apoptosis. Up to now, 5 MCL1 inhibitors drugs are already in the clinical trial and clinical trials. Beta coupling is the we focus on. We use beta coupling as the starting compound. We induce, design, you build, unite, focus on the cytokine, induce build groups, increase activity. We induce hydrophobia, hydrogen bonding, donor decrease activity, and obtain many series ligands. And here we search for you, show you two plate, two copper, chloride, two copper, carbon 9, carbon 8, carbon 9, complex 13, complex 38, and complex 39. And so table 1, you look at the IC of the ligands and complex shown that the ligand here's the some certain and IC50, but our complex, you look at the red line is a better, very long the IC50 values. The copper complex show the highest I thought, then they are again suggest a synergistic effect exists. And another series is nitrate. We change the chloride to nitrate ligand. We obtained another complex and complex 48 and complex 49. As shown in table 2, IC50 or ligands and complex shown that the copper complex show the highest I thought, then they are ligands suggesting a synergistic effect. Also, the structure active in decapitalism, they are have some activity relationship, and the copper complex can inhibit the general cancer cells, whether they inhibit skip the good activity to the MCL1 dependent cells. So in order to answer these questions, we carry out the research on the inhibitor active complex against the MCL1 dependent cells such as the LP1, NCIH 929K6, K562, L35, and MOPA, and the MCL1 independent cells were said by CCKA as before. The research indicate that this copper complex is kept the good selectivity to the MCL1 independent cell, dependent cells. And firstly, we must consider our compound whether abiding to the MCL1. Firstly, we use docking study so that the copper complex tightly binding with P2 binding pocket of the MCL1. And complex completely. Now, as you know, BCL2 family proteins contain many proteins. We must consider our compound selective binding to the MCL1 we use the different method LP, and SPR laser and so on, and different method determine that a compound is keep the very good selected is better than the positive control compound A1210477. And selective also used be found by the other protein. Use also same obtained similarly. And delective binding assay, delective binding assay. Delective binding assays. Okay. Compound, we focus on the compound 38 and 39 and 49, delective binding by the SPR method. The results indicate on compound have a good binding in the KD is reached nanomolar. Use the surface plasma response SPR to invade the kinetic binding for the copper complex with M1. The Italian inhibit very long, the 30.1 nanomolar. Delective binding CD, use another method CD spectrals. And the delective binding ultrafiltration, and use the silent band gene silence and co-immune participation acrylates. And the delective binding experiment in cell, it is very important method. Our research indicate that complex 49 binds to the MC1 protein, and the signaling improve the thermal stability of the protein. We just determine the delective binding, use the small molecular binding to the protein is very easy, but we must consider our compound where they oft target. So we must use the CSA method, and the LD file, and the LD file, the compound is very loud toxic, and more loud than the 6 platons, and they change the body organs waves with research indicated the compounds have a very loud in vivo toxicity. And also we define their pharmacokinetic study indicate that. Also, somebody will ask me the compound where there is in a compress before is it in the blood. So we determine our research indicated our compound is in the blood in form of the complex. And the inhibition of late 39 and 49 have a good inhibition is then higher than the positive compound A121048477. And pathological examination indicate that these compounds, the copper compound, induce skin nucleus in tumor tissues. And the mean survival time mice treated with complex 39 was 40 days, and control is 36 days, 49 days, and which was similar to the secretants 15. So our compound delayed this, increased the survival days, 5 days. And the nitrate compress 49 have seen a similar behavior to the chloride copper compress. The result published in JMC. Part 9. Tubulin, targeting tubulin in the metal compress. Tubulin is the very, very important tubulin is our compound focus on isoapone bond. We introduce a group introduction of threefold affiliate increase the binding activity to the tubulin. And our research, the oxychlorophene, the relative nuclear compass simultaneously targeting microalgae and microtube. The last chemodynamic therapy use the quinoline. Quinoline, the relative specific base to synthesize copper-2 copper compress. This copper-1, copper-2 produce hydroxy radical via Fenton-like reaction by GSH depletion and further induce cancer cell apoptosis via microalgae and ER strikers. And these compounds inhibit the inhibition in vivo. And finally, we get some summary. The coordination regulation effect of Chinese medicine at the ingredients and the metal active centers are further new method to design metal-based and cancer drugs, such as targeting G4 DNA telomerase, targeting protein design, MCL1 tubulin, and induce microalgae apoptosis and autophages targeting Fenton-like reaction, and chemodynamic therapy, ICD effect, autophages, and so on. The anti-tumor activity can be modulated by multiplication, TCMAI, and change the metal centers. And finally, we think our group and our group leader, Professor Liang, and our Pakistan student, and all very good, very large students. Here we just present some published research, and many unpublished research not present here. I hope next workshop we will give more new research to present to everybody. Thank you. My presentation is finished. Thank you, everybody.
Thank you very much, Professor Chen, for your interesting and wonderful talk. With this, the floor is open for question and answers. We do have some questions here for you. Let me read them out for you. Number one is the question, why TCM is not widely accepted worldwide, as TCM has proved a profound drug ability in various disease management. This is from Mr. MD Mahdi Hassan. Over to you, Professor.
Okay. Pardon, please speak slowly. I cannot get a message, get a meaning.
Okay. So the question is, why TCM is not widely accepted worldwide as TCM has proved a profound drug ability in various disease management.
Oh, management. TCM is wide. It is have a very long history in the China, about 3,000 years history, and Guangxi has the rich resource. We focus on TCM, just focus on alkaloids because contained nitrogen and oxygen and donor. This donor can provide long pair electrons coordinate to the metal center, form the coordination covalent bonding. Okay.
Okay, so thank you. There is another question related to it. I would like to know if TCM has been directly used to make metal complexes, or pure ligands were used to make metal complexes.
Sometimes we can obtain from the natural ligand from the plants, and TCM harbors directly binding to the metal center. Sometimes will, according to the medicinal chemistry principles, to design induce different substituents group to increase the binding activity or the targeting activity. So we will synthesis or modifications of natural ligands. Okay.
Thank you. So I think we can take another question. That is, I hope that the metal complexes is within WHO limit for metals, so as to avoid bioconcentration in human if eventually it gets to the clinical trial. So actually, general comment on it.
Okay, it is a very good questions. I have saw the WeChat, the WeChat here. Actually, we determined the metal compass distribute in the different tissue in the mice level, not human body. And the research today, I did not show in the presentation, but in my publication, we reported the research. In the world, a compound mainly accumulated in the tumor cells, and in the lung, kidney, and liver, the accumulation is very large. Okay.
Right, thank you. Professor Chen, as you can see in the comment box that people are saying it was such a nice and informative talk. And with this, I think I can read out another question that is probably somewhat different. What about the solubility concerns of the metal nanoparticles in the biosystem?
Okay, and it's a very good problem questions. Actually, in the metal compass design, we must consider solubility. If the compound have a poor solubility, we cannot carry out any research on the cell level and the in vivo level. So we must select the different anionic group, such as sometimes you use chloride or the nitrate and chloride and other compound, other elements or introduce a water soluble group attached to the ligand. We will obtain suitable solubility complex. But in our group, we did not carry out the nanoparticle research. We just focus on the molecular level, small molecular. We consider the small molecular intelligence the biological target. Thank you.
Thank you very much, Professor. With this, I think, again, I would like to thank you, and I would request Professor Dr. Iqbal Chaudhry for his comments.
Thank you.
Thank you, my dear friend, Professor Zhenfeng Chen, for a wonderful presentation. Your science has always been very interesting and innovative, and you have been very thorough in presenting your science. You have an excellent group of people, and your center is doing very well. So we are very proud to be partner to your institution, and we are very proud to be a friend of you. Thank you very much. Thank you.
Thank you. Thank you. Thank you. Thank you. See you again. I hope this year I can see you in China.
Thank you very much, Professor.
Thank you, everybody. Thank you, everybody. Thank you, every attention. Thank you. Bye-bye.
Thank you very much. Thank you very much, Professor. And with this, we come to the conclusion of our first technical session. And with this, we will move toward a lunch and a prayer break. And we'll start our next session on 1400 hours, Pakistan Standard Time, which means like from 20 minutes from now. So again, we will be gathered here at 1400 hours, Pakistan Standard Time. Thank you very much for joining.
Okay, thank you. Bye-bye.
Okay, ladies and gentlemen, welcome back. We will begin our second session for this online meeting. And we will be beginning with a talk from Professor Dr. Muhammad Saeed. Professor Dr. Muhammad Saeed is from Lahore University of Management Sciences, and the title of his talk is Modern Drug Discovery in Medicinal Chemistry. And with this, I would request Dr. Hina Siddiqui to please introduce and welcome our guest speaker. Over to you, Dr. Hina.
Thank you so much, Kazima, and it's my great pleasure to introduce our colleague and next speaker, Dr. Muhammad Saeed, who is Associate Professor at Department of Chemistry and Chemical Engineering at Lahore University of Management Sciences, which is certainly one of the best institutions in Pakistan. Dr. Muhammad Saeed joined LUMS in 2014 as an Associate Professor. He established the Bioorganic and Medicinal Chemistry group at the Department of Chemistry and Chemical Engineering at LUMS. Originally, he is trained as the synthetic organic chemist, and he has extensive postdoctoral research experience at the interface of chemistry and biology. He is entrusted to address the modern challenges in biological sciences and medicinal chemistry by using the tools of organic chemistry. More specifically, he pursues rational antiviral and anti-cancer drug designing and discovery by modern techniques of the day. Dr. Saeed has received his PhD degree in organic chemistry from University of Tübingen, Germany, under the supervision of Professor Dr. Wolfgang Folter. After accomplishing total synthesis of several natural product and medicinal analog using chirality, topology, and abundant carbohydrates. After PhD, he joined the research group of Professor Irkol, DSc at the Apley Cancer Institute, University of Nebraska Medical Center, Omaha, USA, where he participated as chemical biologist in mechanistic investigation of cancer initiation by estrogen and related carcinogenic compounds. Before joining LUMS, he has also spent some time at the Department of Chemistry, University of Iowa, where he worked as the visiting assistant professor as well as conducting postdoctoral research. He has published around 50 research articles in reputed international journals. He is currently investigating proteases of different viruses like COVID-19, dengue, and HCV as drug target for designing efficient direct-acting antiviral drugs. So with this introduction, I request Dr. Mohammed Saeed to kindly start his lecture.
Thank you very much, Dr. Hina Siddiqui. And first of all, I just want to thank everyone who has organized this wonderful event.
Thank you. Dr. Saab, I'm sorry to interrupt you. Can we have your video on, please?
Okay. Can you see now?
Yes, yes.
Okay. All right. So again, I just want to start off with thanking everyone who has organized this conference and giving a very nice and long introduction, with most of the things was actually refreshing for me as well. I just want to mention one thing that I was lucky to spend some time at HEJ before starting my PhD research in Germany. So I have worked with under the supervision of Dr. Iqbal Chaudhry, and this is an honor for me that I am presenting my work to my alma mater, and hopefully you will enjoy this thing. Okay, so this presentation is a little bit basic in nature. I will not tell today anything about my research. So I will be just talking about how the modern drug discovery or lead discovery is conducted. And this lecture is intended for graduate students and those early career chemists who are actually starting this wonderful endeavor, and they just want to know that from where we can start this drug discovery. So anyone who is interested to know about my research, then I will present tomorrow again, approximately the same time. And over there, my major emphasis will be on the research discovery. Okay, so let's start. All right, so when we talk about a drug, then normally there are two main areas. One is called the centromolecular drug, and other is called the biologics. So biologics, as the name said, it is based on the biological targets like RNA, like protein or peptides. So like, the antibodies. But the centromolecules, these are small molecular species which have the molecular weight in the range of hundreds. That is why it is called a centromolecular compound. So these molecules have played very significant role in the enhancement of life expectancy during the last century. So if you look at the graph, in 1900, the life expectancy at birth rate was about 47.3 years, which was raised to 77.5 years in 2002, which is like 63% overall enhancement in the life expectancy. And if you look very carefully in the region between 1936 and 1942, then we see the bigger enhancement as compared to the year preceding or the years following it. And this era is called the era of antibiotics. when we discovered penicillin, prontosil, chloramphenicol, and tetracycline. So these kind of antibiotics were discovered during that time, and that has enhanced the life expectancy much bigger than the other areas. And if you note one dip of approximately 20 years in 1980, this is a time when Spanish influenza was outbreak, and the life expectancy was dropped significantly. That give us a message, and we are better knowing this message even today, that in the absence of effective chemotherapeutic agent, especially the centromolecular, in the absence of that, the life expectancy is not very much. It is under threat. So especially in COVID era, we know that although there are 6 or 7 vaccines in the clinical usage, but still we are expecting the incident rates are higher, and we are desperately looking for some medicinal agent like drug to treat this infection. Okay, another proof which comes on the importance of this small molecule is the discovery of azidothymidine and another closely related antibiotic, which is the saquinavir, which is a protease inhibitor of HIV. So if you look at this graph, then you can see the rise of death due to HIV. And this rise was pretty constant until we reached the year 1995. So at that time, when these two drugs were clinically used, then we have sudden drop in the death rate. And that indicates that the discovery of the small molecule is the key for us to treat disease and avoid the infection-related deaths. Okay, so overall, this graph showed the current status of FDA approvals. This is USA FDA approval graphs, which show the 2 types of drugs, biologics, which are shown with the green line, and centromolecular, which are shown with the blue graph here. So, and the y-axis, this shows the percentages of FDA approval drugs. And we can see that from 1993 up to 2018, the biggest percentage of FDA approved drugs is based on these centromolecules. Okay. Now, before the beginning, I just want to clarify one important concept, which is more like a technical issue. And that is, as a chemist, we are not looking for the drug per se. We are actually hunting for a lead. So lead, what is lead? Lead is a prototype compound which has very attractive characteristics or feature of a drug, like it have a interesting biological activity, it has very nice IC50 values, but it may contain other undesirable characteristics. And those could be the toxicity associated with that, off-target binding, absorption difficulties, because some chemicals may not be absorbed by stomach. So we have to address this issue. So insolubility and metabolic problems. So these kind of things are still associated with the lead, and they will be removed at the stage which we call the development of drug, and mostly the pharmaceutical companies are doing this kind of refinement. So my topic will be focused on the discovery of lead, which everyone should understand that it is not the final drug. So sometimes we are overexcited, and when we publish some data, then we just sort of claim that we have discovered some drug. So there is still a long journey to make it a drug. Okay, this is the flow chart of the typical or classical drug design. So you always start with the disease pathology. You must set a target, whether it is a cancer or it is a viral infection or it is a cardiovascular disorders or neurological disorders. So you have to select a disease pathology. And then with the help of mechanistic and etiological data, you find out a target. The target, whenever I speak target today, that just means the protein, because most of the drugs are targeted to proteins target, although there are some other targets. But I will specifically mean a protein or a receptor which will serve as the drug target for the designing. So after the validation of drug, validation means that there should be some experiment which tell us that if that particular target is not present in an animal or in a cell culture, then particular phenotypes must disappear. So that is called the validation. And after this, you start your screening. And this is the area where mostly the organic chemists and synthetic chemists, they are involved. So they actually screen drugs either in computational modeling, in silico method, or in vitro cell-based assay, or some phenotypic assays in animal models. And sometimes you can perform only the biochemical assay if you know the protein and their endpoints can be tested in experiment. And at this stage, you normally find which is called the hit. So hit is something which you isolate from a big library of compound. It is like identifying or finding a needle in the straw heck. So this is very important event when you identify an hit. And then sometimes you find several head like you have seen in the talk of Professor Khalid M. Khan, that he has presented many hits which they have identified in the alpha glucosidase activity. So from those structural variants or the different compound, you sort out their hits based on their preference, whether they are commercially available, whether you can synthesize them in a large quantity. So you triage your hits, and then you confirm the hit again with the second time assay, and then you determine the potency of that. And that means you find some intermolecular interaction, how many hydrogen bondings are present, how many van der Waals interactions are happening. So these kind of things are translated into potency. And then the final stage for the lead discovery is the lead optimization. And here you perform without changing much the scaffold of the structure, you just change the substituent and try to enhance the potency and selectivity. So selectivity means that your protein is not binding with other proteins, because for example, if you are targeting protease, then there are several hundred proteases in humans, then you have to find out whether your compound, your hit is not binding to those proteases. And then we have to find the toxicity and try to minimize it. And then we have to find that the pharmacokinetic data and study to find the absorption, distribution, and metabolic fate of your molecule. After your molecule pass this stage, Then it entered the preclinical development, which is mostly the animal studies, and then followed by the clinical study and human. So as a chemist, we are mostly focused on the lead optimization stage. And sometimes if the compound come back from the clinical and preclinical stages, then you can improve the potency, or you can decrease the toxicity as per requirement. All right, so just to give you some the outline of my presentation today, I will talk about the three key requirements for the lead discovery. And the first one is that you should have access to a compound library. And compound, you will see that there are, the chemical space is a lot, it's a huge. And you always try to cover as much chemical space as possible, meaning that your structural diversity should be maximum. And then the large scale synthesis of those compounds should be known, so that at any stage when you need compound in large quantity, then these compounds can be synthesized. And then you have to find the efficient screening assays. So I will talk about two prominent assays which are conducted by pharmaceutical industries all over the world, which is high throughput screening and the fragment-based screening. And these screening give or help us to identify the hit. And these hit, it is, these days it is advised that we should perform the ADME study at this stage. ADME means that we have to check the absorption, we have to see the distribution of the compound in the intact organism like animal. Then we have to find how it is metabolized in the liver and kidney. So how it is excreted, that is the E part. And then toxicity, T. So we have to use different studies. And these days there are computational methods which you can employ. And you have to filter your hit through this either computational method or by experiment, so that the hit are filtered out in the initial state. Because if your compound have toxicity in the end stage, then that will cause the failure of this compound to pass the clinical phases. And then hit after the ADMET filter, it is optimized. Again, optimization means we introduce some electron donor groups, some electron acceptor groups, hydrogen bond donors, and some lipophilic reactors. We have to define this optimization based on the ligand efficiency, which you will see in a little while. Okay. So starting from the library, that what sort of compound should be included in your library? The compound which we can call the drug-like molecules, they actually follow certain rule, which is called the Lipinski rule of 5. So your compound should follow that for the greater success in the drug development discovery stage. And these rules are that the molecular weight of your compound should be less than 500. Okay, so from 250 to 500, so you can choose those kind of molecule. If your molecule is already 600, then it has high tendency to be failed in the later stage of the drug development. Your molecule should have 5 or fewer hydrogen bond donor, meaning that how many hydroxyl group and NH group and sulfur group you can put in your molecule. That should not be more than 5. There should not be more than 10 hydrogen bond acceptor, which are the oxygen and nitrogen atoms in your molecule. Then the compound should be slightly lipophilic. which is measured by log p-values. This value should be less than or equal to 5. These days, they also incorporate the polar surface area, TPSA, which should be less than 140 angstrom square. This is the overall surface potential of your molecule. And this, that have contribution toward the bioavailability and distribution of the molecule in the organism. And then the rotatable bond. So there should not be more than 10 rotatable bonds, because that will introduce a lot of conformational entropy in the molecule. And if molecule is stuck in a conformation which is slightly lower in energy, but it is not the conformation which is required for the biological activity, then this kind of molecule tend to fail in the later stage. Okay, so the molecule which follow these traits, these properties, they are actually estimated to be 10 raised power 60 molecule. And this is a huge number. We do not have that much material in the universe to synthesize all these molecules, and we do not have time to synthesize all these molecules. So we are very much limited to cover the chemical space which can serve for the drug discovery. But at any level, we have to try that the diversity of the structure should be present in the compound library. Okay, so diversity could be the appendage diversity, which means that the basic skeleton of the molecule is same, but the structural moieties, they are different around the molecule. They have different orientation. You can have the diversity or variation in functional group in the molecule. There should be stereochemical diversity. So it should not contain only one type of stereocenter. It should have a different type of stereochemical diversity. And the most important one is the scaffold diversity, meaning that you should not only focus on the sulfonamides or only benzene structure or quinoline structure. You always have to find different structural diversity, structure variation, to find the best lead for your target. So overall, the shape diversity or the scaffold diversity is the key area for the which indicates how diverse is your library. Okay, now the compound source, from where you get all these compounds. So as ever, the natural product are the the best or the very early source of the compounds which serve as the drug, and then dictionary of natural product contain over 200,000 compounds, and many academic and pharmaceutical research are basically focused on the screening of those libraries for different targets. And then we have synthetic compounds. Here you have a lot of diversity. We have several vendors which sell their synthetic analog libraries, which you can buy. For example, Silic inhibitor library. This is very well known vendor who sell this kind of compound. And then medicinal chemistry collections is also a great source for the compounds. And then there are always synthetic chemists who are discovering new compounds collections based on different scaffold or different moiety distribution. And then here, this is the area which I will describe today, is the combinatorial chemistry, which can be performed on solid phase library synthesis or the solution-based library synthesis. Okay, well, if you are interested in the computational screening, then we have a very big data bank, which is called the Zinc database, which have like millions and over 700 plus million compounds. You can download their computational models and screen it virtually by using different docking instruments, docking software, and this data bank also have for 230 million compound which you can purchase. If you find some hit and you want to test them, then this library, this data bank is a very important source. They also have 750 million compound which you can request them to synthesize and purchase them at, for your screening. Okay, now the nature product, we know that nature product has always a great inspiration for the drug discovery, and many important compounds have been discovered from these nature products. For example, pseudoephedrine, which is a stimulant and decongestant, which has been isolated from ephedra vulgaris. Morphine, again analgesic, which has been isolated from the poppy seeds and the stems of this plant. Cocaine is again a stimulant which is isolated from the erythroxyxylum coca, and quinine, which is the first anti-malarial compound which has been isolated from Cinchona officinale. Digoxin is one of the life-saving drugs, which is in the important drug categories in United States. And this compound has been used in various cardiovascular disorders, and this has been isolated from foxgloves plant. And several other antibiotics like penicillin, antimalarial artemisinin, and Taxol, they also have their origin from the nature product. So the nature product should always be given importance when you start your drug discovery effort, and these are synthesized by the enzymatic machinery. And I will describe that many organic synthetic effort has been developed, developed to streamline the synthesis of natural product. Okay, so as I mentioned that the importance of natural product cannot be overstated. Many synthetic chemists have devoted their life to find the total synthesis of these natural product. And the one issue with the total synthesis of natural product is that you can actually make only one compound after a long time and after spending too much resources. So several innovation has been proposed in the first decade of the 21st century. The first one is the diverter total synthesis, which was suggested by Danishevsky, who said that instead of focusing on the target compound or target bioactive natural product, just take one of the advanced intermediate and make a diverse analog of that intermediate, and then screen them for the biological activity. Similarly, the target compound can be verified with respect to structure by keeping the scaffold same and then screen later on. So this strategy, which is called DTS, can be used these days to build the compound library. Wender proposed a novel idea that instead of synthesizing the whole natural product, why not identify the bioactive part of this natural product, which we call it chromophore, pharmacophore. So by comparing different natural product with the similar biological activity, you can come up with the novel pharmacophore and then build the library which is entirely based on these pharmacophore. So this is called the function-oriented synthesis. The Schreiber proposed another idea which introduced the skeletal diversity in the library. So he just started with the simple starting material and then introduced the structural variation by performing different type of reaction in the beginning, and then each structural diverse molecule can be further diversified by introducing different substituent. So that way you can build a library using the DOS. And lastly, very recently, Abbasov have proposed a pharmacophore directed retrosynthesis, which means that you take one bioactive natural product and isolate different pharmacophore and drive your retrosynthetic analysis, which isolate or which end up with that proposed pharmacophore, and then you do the structural variation synthesis of analog of that pharmacophore in the end. And that will give you the library of that compound. Okay, one strategy which is called the biological oriented synthesis or BIOS, that is based on the structural conservatism in the protein and in the natural product. We know that the proteins are actually, they have their three-dimensional structure. When the beta sheet and alpha helices, they fold on each other and then they develop a certain type of fold or crevices, which are actually used by different drugs for the inhibitory study. And it is known that the fold of protein is not the sequence dependent. It is actually a little bit higher in higher in well in the hierarchy, and many proteins with the different with the low sequence homology, they can end up with the similar fold. So there is a conservation of fold in the protein. And similarly, the skeleton or scaffold of molecules, they are also conserved with respect to their basic skeleton. Okay, and another important thing to note is that all natural products are synthesized by enzymatic machinery. So these are molecules which have already interacted with the biosynthetic enzymes, and they are actually well oriented to bind at a particular fold or particular fold of the protein. So the proteins, they come up with a little number or small number of fold as compared to total number of protein, and their scaffold, they have binding interaction for a particular type of fold. However, the interaction or the potency or the binding of this of the molecule in a similar fold, that depend on the orientation of side chain. For example, if these two scaffolds can bind in a protein, then only one protein which have the properly oriented substituent which can make interaction with the side chain of the protein, they will give the better binding interaction. They will be more potent than the other type of scaffold. So the interaction or the shape of the scaffold that help to bind at the fold of the protein, but the actual binding take place when the residue and substituent, they are complementary to each other. All right, so the other technique which can be used to build the library is the combinatorial library. And this technique actually emerged when high throughput screening was taking place in the industrial setup. And they always need large amount of large number of compound to screen. So this combinatorial chemistry that help in that direction. So combinatorial chemistry give rise different type of arch product. For example, you can build polymers with different units which are A, B, C. So they have different position in the polymer structure. You can build carbocycles and heterocycles where the position or the point of diversification can be located at A, B, and C sites, or you can build the binary compound using combinatorial chemistry. So these type of products are easily built by using the combinatorial chemistry. So combinatorial chemistry come up in two different modalities. One, the well-known or the old one, is the mix split strategy. In this strategy, you actually split, for example, a solid sport in the different vessels, and in each vessel you react one starting material, one building block at a time in different vessel. For example, in vessel A, you react with or bind your building block A. In B, you build a compound with the B attached on the solid sport, and in C, you have the compound C, which is attached to the solid sport. And then you mix all these things, A, B, C, you mix them all and then split again into the same number of vessels. And now in each vessel, you will have not only A, but all other, the starting material will be available. And then you react with, again, separately with A. So if your initial compound contain A, then you have the AA combination. And if your initial compound has a B building block, then it will make the BA, and similarly you will build the CA component here. And in the vessel where you react with B, then you have the alternative combination, and in C you have entirely different combination. And in the third stage, again you mix them and then split in the equal number of vessel, and then you react with the component A, B, C again, and then you can introduce the diversity in the scaffold. The second strategy is called the permutated parallel synthesis. That is mostly the well plate model, that you have a multi-well plate in which you have a different building block A from A1 to AN, and different building block B from B1 to BN, and different building block for C components. So these A, B, C, they are different compounds which have a different functional group. If you recall the talk of Professor Dr. Khalid, then you will see that there are some reactions which are multi-component, and you can build those kind of library in the well plate model. So here you first fill all well with the compound A, for example, and plate B is filled with the compound B, and so on, so forth. You fill all plates based on different type of A. And then in the columns, you put the B1 component in the columns, and in the column wise, you distribute all B1 component, and in the row, you distribute the C component or the C building block, and in each plate and in each well, you will have a different type of compound, and within a short time, you will have the library of 1,000 compounds uh, built on the combinatorial chemistry. Okay, here is one example in which 1,4-benzodiazepine derivative was synthesized using the solid phase parallel synthesis. The sequence is simple. I will not go into detail. Only I will describe that the benzophenone, aminobenzophenone, which is protected at the N position, that is deprotected by piperazine, and then free an amino group react with a protected amino group to make the amide bond, and then you do the deprotection again, and then the free amine react on this ketone functionality to make a cyclic structure, cyclic imine. Then using a base and reacting with alkylating agent, you furnish the target 1,4-benzodiazepine, which is cleaved from the solid spot and ready for the screening. And by just using 2 amino benzoquinone and benzophenone and 12 amino acids and 8 different alkylating agents, you can build a library of 192 compounds within 2 or 3 days. All right. So if you are still looking for more reactions to perform to build the library, then the last option is the multi-component reactions. For example, Ugi reaction is well known for the synthesizing dipeptide type molecules, especially in the protease area. This is very important reaction. Where 4 different reactants, aldehyde, amine, isocyanide, and carboxylic acid, they are reacted in a one-pot reaction, and they give the final product. And by varying the substituent as R1, R2, R3, and R4 position, you can build different type of analog. And Bignelli reactions can be conducted to make this this pyrimidine type of molecule, and then Greco reaction can be performed by aldehyde, aniline, and alkene type starting material. So many multi-component reactions are available which can give rise to the libraries of these compounds. Now, let's switch our gear and just talk about the screening method. So as I mentioned before, that when I talk about the target, that particular mean the protein structure. So based on the target, we can do 2 broad areas of drug designs. If we know the 3-dimensional structure of protein, then we can use the structure-based drug design, which is called SBDD. And if we don't know the structure of 3-dimensional structure of the protein, then ligand-based drug design is the option. So this ligand-based drug design is the mostly used technique, because many at times we don't know the three-dimensional structures of the protein, especially the proteins which are present at the cell membrane or plasma membrane. Okay, so within each category, if we know the structure of ligand, the natural ligand which bind to that protein, if that is also not known, then the best option is to do the high throughput screening or the fragment based screening using the cell culture models, or even animal models. And here the important area which we are looking for is the phenotypic changes. In the ligand-based drug design, if we know the structure of ligand, the ligand which bind to the protein, then we can do the pharmacophore modeling or similarity search, meaning that we take the structure of the natural ligand and try to diversify it by using different synthetic method. So now if your protein structure is known, but the ligand is not known, then we can do the de novo design. We can screen different libraries of synthetic compound, or even do the virtual screening and identify the hit, and then we can try to determine the three-dimensional structure or the co-crystal structure of this compound. And then lastly, if the structure of protein and the ligand are known, this is the area which is called the structure-based design, and mostly the co-crystals of ligand in the protein are very helpful to discover, to screen the library of the compound. Okay, so the high throughput screening, they have a different features, which are the first one is that it is a very rapid screening method. You can automate this method using robotics, liquid handling systems. It can be managed with sensitive detection systems, and it can come up with the different plate models, well models like 96 to the very high or ultra high throughput screening, which contain thousands of well per microtiter plate. Different biological assays are available in this model. You can perform biochemical assays. You can do the in vitro assay, cell-based assay, or the whole organism can also be tested using the high throughput screening. Then we have the random screening, the library of the drug-like molecules can be screened, or the targeted screening when you already know some structure, some hint of the design of your molecule, then you can perform the targeted screening. And the one of the technique which is frequently used these days is the fragment-based screening. And the difference between high throughput screening and fragment-based screening is only the low molecular weight compound in the fragment. Here, the compounds are mostly equal or less than 300 molecular weight. So the screening and the diversity is smaller as compared to the high throughput screening. In high throughput screening, you have to screen millions of compounds to find the hit, whereas in the fragment screening, you can come up with the lead by screening thousands of molecules, not in the millions. So fragment-based drug discovery is in practice since last 20 years. It has helped us to discover 4 FDA-approved drugs, which are shown here, and 40 other compounds are in clinical trials. The features of fragment-based drug discoveries they are mostly based on the low molecular weight fragments instead of the bigger or the larger molecules. The hit selection is based on the ligand efficiency, not the potency. Here in the fragment-based drug discovery, the focus is not always to have the most potent compound. It is the most ligand efficient compound. I will describe this term in the next slide. There are different methods which can be applied for the optimization of potency, and I will touch this technique also in the end. For screening, we have different modalities. You can do the X-ray crystallography and NMR-based method. In NMR, we have variety. If your molecule contain fluorine, then you can perform the F19-based NMR screening. Or two important techniques, which are the ligand observed technique, is the saturated transfer difference STD NMR and the water log C technique, which is used to screen the library of compound in high throughput fashion, or the fragment based library. If your protein is labeled at the nitrogen, labeled with nitrogen 15, then you can perform the two-dimensional or three-dimensional technique like HSQC or the other advanced NOE or NOSI techniques to observe the conformational changes in protein when the ligand bind at the binding site. Other technique which are used is the SPR, sulfur plasmon resonance, and isothermal calorimetry and microscale thermophoresis. I will talk about these techniques in the next slide. All right, so what is the principle of fragment-based screening? So here first you screen a library and you find the hit. And this hit is actually evaluated based on the intermolecular interaction with the receptor. And then this fragment is increased in the complexity by introducing different structural moiety. So normally they elaborate the fragment by introducing 6 or less than 6 heavy atoms at one time. So with this performance, you can grow your fragment and eventually end up with a potent drug. So this cartoon describes the basic or the fundamental of the fragment-based discovery. So there are two advantages. The first, you don't need to screen a huge library to come up with the same potent molecule. In HTS, you have to screen from 100,000 to 1,000,000 compounds to find the better hits. Whereas in the fragment-based screening, you have only a small number of fragments which can give the same potency or even better potency compound. So the second thing, second advantage is that the interaction of molecule which you discovered by HTS may not have the better ligand efficiency. Ligand efficiency is the interaction per atom of the molecule. So if this molecule have, let's say, 20 different atoms, then how many atoms are interacted with the protein, that is measured in the ligand efficiency. So the fragment-based hit, they have higher ligand efficiency because most of the atoms are interacted with the ligand, and the more the number of interaction with the fragment, better will be the potency of the molecule. So the fragments are ligand the atom efficient binder as compared to the hit which are discovered through HTS. Okay, so for fragment-based screening, we normally look for the binding interaction, which are normally in 0.1 to 10 millimolar range. So normally they are not great in the beginning, but they are improved in the later stage. The sensitive biophysical screening method must be used. For example, NMR, X-ray crystallography, or ICT. These methods should be used to screen these hits, because the conventional the biochemical assay, they normally fail, or they do not give the better result when you perform the fragment-based hit. Compared to the typical HTS bioassay, the higher concentration of fragment are used, and that is obvious, because if the binding interaction is not potent, then you have to start with the very high concentration in the beginning. The fragments are chosen because of their good aqueous solubility. So the fragment, they contain mostly the polar group in the beginning, and they enhance the water solubility, and that has the advantage because we will get very few false positive result due to aggregation of the molecule, and this is a very common problem in the HTS screening. The important biophysical method which are used, I have already described them, and I will describe them in detail here. The first method, which is very useful, is the fluorescent-based thermal shift assay. So here the idea is that when target is isolated, if your protein is empty, then it will melt at a lower temperature as compared to when the protein is bind with the fragment. So binding of fragment with the protein, that will increase the melting temperature of your protein, and the difference will tell the binding interaction. So based on that difference, you can select your hit. A molecular spectroscopy can be used because if your target is empty, then it has a lower molecular weight, and the molecular weight will be increased when the fragment is bound at the binding site. NMR water log C technique is based on the magnetization transfer from water and the ligand to the protein, and vice versa, and the signals are enhanced due to some NOE effect, and the ligand which have interaction, or which bind with the receptor, they will produce this water log C signal, and those fragments which do not bind, they will not produce the signal. SPR technique is based on binding the target on a solid chip, and letting the solution of different compound to flow over it. The compound which bind with the receptor, they will increase some physical phenomena like refractive index, which will be displayed as the binding indication. So here, the greater the binding, better is the response in the SPR, and from there you can select your hit based on the better signal from SPR. X-ray crystallography is based on the co-crystallization of a fragment with the protein. And lastly, the in silico study, are the docking, that is well-known method these days, which give the binding interactions and the binding poses of the molecules, which can help to identify these the fragment. Okay, so after the identification of fragment, the fragment is elaborated, and I will show three different methods in which the fragment can be grown further to enhance the potency toward the drug lead discovery. So first method is called the fragment merging. Here you have the co-crystal structure of a fragment and match it with the known co-crystal structure with any other compound, and by matching the by overlapping the both structures by different computational software, you can predict that at what stage, at which site the molecule can be grown to enhance the activity. For example, uh, in this slide, you can see the fragment which have the interaction with 20 micromolar concentration, and a known alkaloid which have a co-crystal structure and that have the activity in 32 nanomolar. And when you merge them, then we identify the cavity that this molecule can be grown in the nitrogen side. And when the molecule is substituted at this nitrogen, then the overall the potency of the fragment is increased from 20 micromolar to 350 nanomolar. The second thing is the fragment linking. In this method, we identify the binding in different pockets of the protein and then link them, these fragments together using a linker of appropriate site, which have this you exemplified with this molecule with IC50 of 330 micromolar and 100 micromolar, which are very low potency bindings. But when they are linked together, then the potency is increased to 1.4 nanomolar due to the entropic constraint. And the third technique is the fragment growing. Here the fragment is arbitrarily grown in any site, and by checking the activity at different stages, we can come up with the molecule which have the better potency. So here is the example of a molecule, a drug, which is AT9283, which was developed through this fragment enhancing fragment growing method. So here, this is the fragment which have a very low potency, but it has very high ligand efficiency, 0.59, which have different IC50 against different aurora proteins. And when this fragment is grown at this site, then the potency is increased from 0.9 micromolar to 100 nanomolar. And by further refining or reducing some number of carbons, so that the ligand efficiency should be enhanced. So the ligand efficiency here is not good, but when you reduce this ring size from 6-member to 3-member, then ligand efficiency is increased, giving rise to the IC50 and 3 nanomolar range. All right, so here is the comparison between the conventional high throughput screening and fragment-based approach. And in HTS, the emphasis is on the potency, whereas in fragment-based, the emphasis is given on the efficiency, not the potency, so which is improved in the later stage. So here, the hundreds or thousands of compounds should be screened to find the lead, whereas very few hundred compounds are sufficient to discover the lead. The molecular ranges are higher in HTS, whereas it is a lower molecular range in the fragment base. Hit activity is low micromolar to nanomolar. Normally, this is what we call the hit that should display the activity in this region. Whereas for fragment based, the hit normally show activity in millimolar to low micromolar range. And here the ligand efficiency is low, whereas the hits are identified by the higher ligand efficiency when you are doing the fragment-based drug discovery. All right, so HTS is the extensive, it requires extensive infrastructure. You need the robotic equipment, you need a huge library of compound. You need big data processing systems, and you need to do a lot of chemistry, whereas fragment-based approach is not very resource intensive. However, we need the expertise and knowledge in the protein structure and molecular interaction to be successful in the fragment-based approach. So lastly, if you have discovered the hit, then how you can improve the drug likeness in the molecule. So there are different methods. So first you do the you confirm the hit by repeating the activity, and then you synthesize the different analogs and conduct the SAR study to find to do the hit evolution. And then sometimes you have to do the bioisosteres replacement to improve some pharmacokinetic data. And interestingly, sometimes if you find the hit from high throughput screening and you take only the structural scaffold of that molecule and convert them in different fragments, and then you can do the fragment-based techniques to improve or to discover the hit to lead stage. And if you have the fragment-based hit, then you can improve, you can find the lead by joining or expanding the fragment in different method as I have described earlier, or by joining different fragment in different fashion. Yeah, so that's it. Actually, I stop here because I think I am little over than the allotted time.
Saeed, I'm sorry to interrupt you in between. Actually, we are like behind the time. So can we just quickly finish?
Yeah, I'm happy to answer any question if you have. Otherwise, we can continue with the second presentation.
Can you open the chat box, please? We can have a look at the chat box for the questions. We can have one or two quick questions if we have. Okay, more of appreciation and the request to share the presentation. With this, I would like to thank you, Dr. Saeed, for very informative lecture, and looking forward to your academic scientific lecture tomorrow, inshallah. Thank you so much. With this, we can move to our next talk of the day, which is our last but not the least. And to introduce us with today's speaker, I request my dear teacher, Professor Dr. Mohammad Iqbal Chaudhry Sahib, to kindly introduce us with our next speaker, who is Professor Dr. Mustafa Husain.
Hello, can you hear me?
Yes, Professor, very well.
Okay. Okay, I apologize. I think I messed up the time difference. Since you guys are 3, I thought you guys are ahead of us, but I was wrong. Sorry, I messed up with the time difference. Sorry, I apologize again. So let me just. You guys can hear me, no problem, right?
Professor Iqbal would like to introduce his brother. So,
sure.
Yeah, sir,
So
please.
please. Yes.
Mustafa, I don't want to miss the opportunity of introducing someone who is so close to my heart.
Thank you.
So
Thank you.
give me an honor of introducing very dear brother, a renowned professor, someone who combined the skills of organic chemistry, medicinal chemistry, and industrial experiences of developing drug in real life. So Professor Dr. Mustafa Gozel, he is affiliated as the Director of Drug Discovery and Development Research Center as the Istanbul Medipol University, which is a very prestigious organization. Professor Gozel has worked tirelessly in various fields. During the pandemic, he has been extremely busy in the development of many new therapeutics. In fact, his contribution has been recognized both nationally and internationally. He was graduated from Hacettepe University School of Engineering, Ankara, Turkey, with a BSc degree in chemistry in 1987 and completed his MSc and PhD degrees in organic and medicinal chemistry at the Clemson University, USA in 1996 and 2001. He worked as organic lab coordinator at Northeastern University and later joined to Arcueil Incorporated as a senior synthetic organic chemist upon his graduation. So he has actually worked with the top industry. He then joined Transtech Pharmaceutical Incorporated in High Point, North Carolina, and assumed various positions in medicinal chemistry at department between 2001 and 2014. So he brings with him a long experience of working in industrial setup. In 2014, he started working as an assistant professor at International School of Medicine in Department of Medical Chemistry, Medical Pharmacology at Istanbul Medipol University. In 2017, he was promoted to an associate professor in medical pharmacology, acting as a chair. In 2019, he was appointed as the department chair of molecular medicine and biotechnology. In January 2020, he then became the director of the Drug Discovery and Development Research Center, a newly established health science and technology research institute at that university. He has a tremendous contribution in solving real life problems. During the pandemic, his contribution has been widely recognized. He moved from laboratory discovery to industrial production and has made a seminal contribution in Turkish national struggle against the pandemic. In fact, some of his contributions have been recognized at the highest level, including the president of Turkish Republic. He is a great supporter of science all around the world. He has a global vision, and he has been coordinating, collaborating with people in different continents. With this, I would like to invite very dear Professor Dr. Mustafa Gozel for his presentation.
My dear friend, Professor Iqbal, I want to sincerely thank you for inviting us, and also Hina for this opportunity to give our recent studies. And I wish all of my dear Pakistani friends and Eid Mubarak. I know, although we passed just week, but I hope you had a prosperous and nice Eid and last week. So thank you for inviting me again, and I sincerely apologize. I messed up with the time difference. I thought my time was at 5 p.m. in Turkish time, but it was 1 p.m. on Turkish time because I messed up the time. Anyway, so thank you again. You guys can hear me well, no problem.
We can hear you, but I would appreciate if we can see you as well, sir. Turn on your video.
Camera is on.
Yeah.
You guys don't see me?
No.
Okay. I thought I see myself. I don't know. If I stop share, you guys can see me now?
No, sir, only your picture is there.
Now, how about now?
Yeah, it's
Yeah.
fine now.
Now it's fine, all right? Okay, all right. So let me share again the presentation. Now you can see me both and my presentation, right, Ina? No problem.
Yep.
Okay, all right. So, okay, thank you again for this nice invitation. My seminar outline, I put the timer. I don't want to talk too much, but the seminar outline of my talk will be like a little bit current situation analysis where we are, especially give you the feedback of drug discovery and development. I know last time I talked about the industrial production of favipiravir, which is the favipiravir, the trade name in Turkey. I will highlight just a glance at it, and then later on move on to natural medicine for COVID-19, which is our recent accomplishments in the COVID-19 treatment, which we are about to submit. Actually, we submitted the manuscript to phytochemical analysis. And then we will talk about a little bit other projects, just with the fast track acknowledgements, and the conclusion remarks will be, then I will try to speak around 40-45 minutes, if that's okay, Hina. Then if there are some questions, dying questions, please feel free to ask. You can anytime interrupt me if there is really questions related to the subject. So I want this at least lecture as much as possible, more interactive. So as Professor Iqbal mentioned, this was the first project that we successfully finished, completed. This was the title of the project, was the development of the drugs and the raw materials with the new synthesis methods and the formulations to reduce external dependency against pandemic coronavirus. So without going to that topic, I want to give you the feedback. There was a survey which was done last 2 years back. And this is the link on the bottom, you can see. Basically, who do we trust? Do we trust the scientists, doctors, lecturers, police? You know, which professional jobs mostly? This was the survey which was done worldwide, in Turkey as well. I don't know if it was done in Pakistan. So mostly, overall, people trust scientists and doctors, and then later on, teachers and the armed forces. And this was the countrywise distribution. Mostly, again, scientists lead the way, then the doctors and the teachers. After Russia, Argentina, and Mexico, Turkey was the fourth country on earth. People trust mostly scientists. So, and here are the basically the distribution among the, again, scientists, 12 countries, again, number one. And the doctors, I think 8 countries, number one, including Sweden, Australia. Interestingly, teachers are the most trusted in Brazil and the USA. Needless to say, in all countries where they asked, politicians, I don't want to interrupt or to deal with the politicians, consider it the untrustworthy profession, because the people, the promises sometimes may not be kept, as you know. So I have this bad habit, pictures with the Nobel Prize winners. I don't know if it is a good habit or a bad habit. Not that I'm gonna win one day Nobel Prize, but it's just a habit which I would like to share it with you guys, especially with the young researchers, to give them at least the passion to become passionate in science. So Aziz Sancar, as you know, the professor, he's a professor at UNC Chapel Hill, won the Nobel Prize. This is the only Turkish American scientist who won the Nobel Prize in science, basically. Then E.J. Corey, Harvard professor in 1990 when I was doing my PhDs. 2001, Sharpless, who won the Nobel Prize in 2001. Actually, this picture was taken before he won the Nobel Prize. I was very happy because during my PhD studies, I was following his footsteps because I was doing asymmetric hydroxylation, dehydroxylation studies. Not only you get the EEs, the yields are actually accurate or identical with his research, you get the EEs identical with his research. So I was right. He was my idol. So I emailed him. And he said, I congratulated him and I wished him the best. And he sent me email back, Mustafa, thank you for your email. I wish you success in your career. So these guys are also very humble. This is what I want to say. So Negishi, 2010 Nobel Prize winner. Brian Kobilka, Stanford professor, 2012 Nobel Prize winner. And 2008 Nobel Prize winner, Martin Chalfie, who actually was the pioneer of developing GFP, green fluorescent protein, as you know. And this is the year, maybe you're gonna catch my, how old I am, my age. This is the first time when I entered the lab, we were trying to actually isolate or extract the essence of the orange, peeling the orange on the orange peels, basically. Fragrance of the orange, basically. And the right picture is the when I was doing a in vivo study with diabetes drug development. So drug discovery and development, as you know, is lengthy. It's very difficult. It requires really extensive infrastructure and resources. But as you know, it's not impossible. And I wish to visit one day to Professor Iqbal's and Hina's research infrastructure, which I would like to collaborate in future. But with a team effort, it is possible. I know we are in the eastern part of the world, still developing world, but if we tie and bring this, our strengths together, we can do it. As I don't want to give in the detail, but you know, mostly I concentrate myself in the dark blue area, which is the pre-discovery or discovery in vivo pharmacology, ADME, PK, PD, pharmacokinetic and pharmacodynamic studies. So it's a medicinal chemist. If you bring a compound in IND, investigation of the drug, for us, actually, that our job is bring one more molecule, which we call backup compound or a lead compound, same efficacy and same potency and same toxicologic profile. Then for us, that project is done. So then the destiny of the project now in the hand of the human trials, as you know. So and the scientific disciplines and the development capabilities needed for drug discovery is, you know, is this. All the scientists and experts has to become together, to stick together, basically to develop or discover novel molecules. That is the handicap sometimes I see in our culture and in our region. So that we actually should stick together and study together in order to develop new molecules. So as you know, the drug discovery, you have to have a first target, the identified target for the intended therapeutic site or action. Then you have to catch a fish, which I call this, you have to basically find a molecule either that activates or inhibits or blocks or potentiates that intended target. And then after that, you basically optimize that lead compound until you reach the big fish, basically. This is what I look at it. It's lengthy. In my career, probably I synthesized 12,000 compounds. Only 12 of them became drug candidates. Only 1,000 to 1. So imagine that. It's so as a scientist, we should realize that we cannot do it ourselves. As an academician now, I worked in the industry, as Professor Iqbal mentioned. Now I'm a fresh in academic. but we can do a lot of studies in academia, rare cancer, infectious diseases, neurodegenerative disorders. We can do cell-based assays, some enzyme activations or inhibitions. Bottom line, we have to partner with pharma. This is what I want to emphasize. So you can do assay development, heat discovery, lead optimization, animal model efficacy, publication, training, the young scientists, PhDs and postdocs. But at the end of the line, I want to emphasize, I mean, making one molecule, 1 milligram is much more easier than making 1 ton. So when you become an industry level, it's a whole different scenario. So with that in mind, federal and industry roles in the R&D is really complementary. So governments usually should support the basic research and some sort translation research, but minimize the clinical research. Mostly the clinical research should be supported by the basically the pharma or the global pharma or private sector or investors, angel investors, because it's lengthy, it's costly. This is also done, or this exercise is done in the US. It can be done in the Pakistan as well as Turkey. I like this slide also. This is the global companies. At least give you the feedback how we should strategize ourselves. So 15 years of R&D expenses and how many molecules they brought to market. When you, for example, give an example of the AstraZeneca, for example, spend around $58 billion in 15 years, but can only bring 5 molecules. Whereas Amgen, they spend half of the other company, but they bring 9 molecules. But we should also consider their their revenue, right? How much money they are making back, because that's also important aspects of it. But if you're a global company like Pfizer, right now we know that BioNTech and Pfizer making this vaccination. So if you are a global company, you can spend billions of dollars for R&D. But if you are a country like in Turkey or in Pakistan, you're much more limited. So what I mean to say, we have to focus on certain therapeutic areas, certain expertise areas, that what we can gain and what we can basically bring on the table. That's what I mean. Okay. So another interesting fact that I want to highlight: 90% of the molecules that we bring into the clinic, unfortunately, or phase 2, fail reach to market, unfortunately. So that's why we have to strategize ourselves. We have to make it in a way that it has to be a win-win situation. Bottom line, we need to reach the ultimate goal. As we know, because average cost of to bring the new drug to market is almost $2.6 billion right now. I think people are saying right now is almost $3 billion. I mean, if you don't have your $3 billion, so if you have limited resources, then you have to go strategize yourself. Less animals, less molecule, and less clinical trials to finalize yourself. So as a medicinal chemist, we should realize that every molecules we make do not become medicines, because the disease targets are too complex. There are some adverse reactions, absorptions are poor. It doesn't go to the brain. For example, you make a molecule, it doesn't penetrate the blood-brain barrier. All of these things can play a role. And I gave you an example on my side. In my hands, I probably synthesize around 12,000 compounds, but only 10 of them or 12 of them, only a dozen of them became a drug candidate, not drug yet, don't forget. So this is an important aspect. We should realize that. And without bothering you too much, and I want to give you an example of one project that, as Professor Iqbal mentioned, this was a TÜBİTAK supported project. When the corona hit the world, of course, it became a pandemic in March. So in Turkey, before even March, in February, we said, okay, which medicines we should pursue. Because as you know, even the mask and disinfectants were short in most of the countries. They were in need. So, and also starting materials, APIs, most of the countries, even in China and India, it was very hard to bring the compounds or the APIs to country. So we said, in order to help our country, what can we do? These are the three medicines that were probably, as you know, all of them, were actually the pioneers of the corona treatment. Favipiravir is the first one. Then the anti-malaria medicine, hydroxychloroquine, although now HCQ is not first line of treatment in Turkey right now. Remdesivir, as you know, first drug that approved by the FDA in somewhere in 2020, as you most of you guys know. So although this was a patented, protected, but that doesn't mean that we cannot synthesize and we cannot make a lab research out of it. Of course, you cannot sell, you cannot manufacture it because it is patented molecule. But the first two you can. This is what we did basically, because favipiravir was the first line in treatment in China and in Japan. It was discovered by Toyama Chemicals, and trade name was the Avigan, 200 milligram tablets, and it was approved by Turkey and some other countries for the treatment of COVID-19. And the first dose was the 1600 milligram, and then follow up next days. What was the effect? It was effective for RNA viruses, other than the corona. Of course, before it was used for basically common cold and the flu. And what is the mechanism of action? It's basically RNA-dependent polymerase inhibitor. It blocks the proliferation of the basically virus. Of course, there are side effects. It's teratogenic and embryotoxic. That's why the pregnant woman cannot use this, and the partner cannot use it. It will harm the baby. So it's a product. It tautomerizes with the help of enzymes. It phosphorylates. Then it becomes active form is T704 active form in the body. So there are some protein binding database, and this is the one that I found. This is the hypoxanthine guanine phosphoryl transferase, the binding affinity of the enzymes, but there is not much study of the for the main protease and the other enzymes, how it binds. How is it synthesized? This is the synthesis of favipiravir. I don't want to bore you with the favipiravir, which I want to emphasize most natural products. So it's 7 or 8 steps of synthesis. We focus mostly, we brought from the ester to compound to dichlorocompound, which is 6. And then after that, we wanted to emphasize and increase our yield from 7.1 to 7.4, which is the favipiravir, 7.3. So that was our effort mostly, one-part synthesis without isolating the intermediates. So our goal was how can we develop the process at the university level or a small scale and apply that research or knowledge to industrial scale. That was our goal, and optimize that method to industrial level. That was what we did. And these are the intermediates that we actually, I wanted to emphasize this, at least the visuals that you guys can see, the melting points and the yields for each intermediates. We are about to, by the way, submit this process that we developed for this favipiravir synthesis. And then we started actually work on the optimizing the process. How can we increase the yield, and also without isolating the intermediates, how can we increase and develop different pathways, and in order to increase the yield. As you know, in industry, even 5% of the increase of the yield can mean a lot. It means you save huge money because of the yields increase. So this is what we spent basically in the last year. Method optimization, of course, even right now, still currently ongoing in the industrial level. But in the lab level, we basically successfully generated favipiravir in a large scale. These are the proof that we basically by HPLC and MR and compare with the authentic materials. These are the results with the comparison: proton, carbon 13, and fluorine 19, NMRs with the authentic material. And then we increase the yield to about 45 to 50% right now by isolation method, extraction, filtration, and crystallization. Of course, in industry, you don't want to do columns or you don't want to do separations. You need easier to finalize your final product. And in July 2020, I apologize, this slide is Turkish, but you probably understand this is the basically license from the Minister of Health that we are approved to manufacture and to sell, or not to give it to the Minister of Health in order to use it for COVID-19 treatment. So as a favipiravir results, we following the 8 steps route, we finally were able to synthesize in a lab and industrial scale was completed in the domestic force and the facilities. The structure of the target compound was determined by known analysis like HPLC, LCMS, NMR, and optimization studies have been completed for each step. And the monthly production capacity right now reach about 3-4 tons. And its industrial scale production alternative synthesis methodology is still, of course, ongoing. So there are some alternatives. As Professor Iqbal mentioned, President Erdogan was accepted actually congratulated us to actually finish this within the 40 days. So we were able to discuss the scientific perspective of our country, how can we improve and increase the scientific basically area of our research capabilities in Turkey. So with that in mind, I sincerely thank President Erdogan accepting us and also basically taking ownership of these projects. So this was the project. The crew during that time, we were only 12 people. So within 4 days, we were able to bring this compound in no time. Right now, we gave around probably 3 to 4 million boxes to Minister of Health, so that we don't dependent of this API or this medicine anymore. Without losing too much time, I'm looking at the time right now. We are 26 minutes, so I think we have time, right, Ina? So natural remedies for COVID-19 prevention and treatment, which I will highlight. As you all know, when the COVID hit the world, people also, we are good, right, Ina? Yes, okay.
Yes, yes,
So
yes.
Okay, thank you. When the COVID hit the world, people started searching also some natural products, even prevention, because before even you become a COVID-19, you can use some natural medicines, as you can see here, some flavonoids, some benzonoids, or some fatty acids, even lycopene, for example, or linolene, some terpenoids. These are the ones that they call the hyperfoods to fight for COVID-19, even before basically you get infected. So as you know, there are huge studies right now going on for natural products for COVID-19, and some of them I want to highlight here. For example, the gingerbread or gingerols, hypericin, allicin from the garlic, and Bisalin, the list goes on, and especially this thymoquinone from Nijale sativa. Right now, we are also trying to start a phase 2 study and phase 3 study in US and as well as in Turkey with this GMP production. Inshallah, we will start soon. So there are some natural products, as you know, and here are the examples. There's epicollagen galates, genistein, indole containing molecules like indole trimethanol, alpha tocopherol, naringenin, gallic acid. And these are the natural products that we can probably think of it. Of course, there are more. And here are the examples again. Some alkaloids for SARS-CoV-2 treatment. Again, leucorin, amyretin, tylorcorin. I don't want to highlight most of them, but I think you get the idea that actually there are a lot of flavonoids, chalcones, biflavonoids, tannins, glycosylated flavonoids for the treatment of COVID-19, SARS-CoV-2. And as I mentioned, black cumin. I don't know Pakistani name for this, but this is, as you know, Prophet Muhammad Sallallahu Alaihi Wasallam, as you know, mentioned this. This has a treatment for a lot of things. So the chemical content of this thymoquinone also is believed to be strong in the treatment of COVID-19. Turmeric is another one. Curcumin is also still under the investigation. Oregano oil, thymol, and carbachol. As I mentioned a minute ago, the garlic and the garlic allicin, and this EGCG, another important molecule. Quercetin, and quercetin is also, when you look at the probably FDA, and there are a lot of molecules for the treatment of natural molecules or natural remedies for the treatment of COVID-19. Another one is boswellic acid or boswellia, artemisinin annua, which I will highlight a minute later, curcuma or curcuma longa, and the list goes on basically. So our second project, which I will highlight without bothering you too much, I will finish it in 5-10 minutes, is the isolation and synthesis of natural molecule, the analogs, in vitro in vivo investigation of antiviral effects against COVID-19. So we chose artemisinin annua. Why? Because this was grown in the Bursa region, which very close to Istanbul. We were actually fortunate to get 5 kilos of this freshly grown Artemisia annua. And then, so we immediately started isolation, extraction, and characterization studies. So this was the first plant that we chose for the studies of COVID-19 treatment. As you know, Madagascar president, he claimed that herbal tea remedy for coronavirus, and he drank actually Artemisia annua herbal tea. This was actually on media. Probably some of you are aware of it. So another important milestone I want to highlight about Artemisia annua is, as you know, in 2015, Artemisia was used by Chinese scientists for the treatment of Vietnam War and has been tested in laboratories for a long time. And a Chinese doctor, Tu Yu, won the Nobel Prize in medicine for the discovering and purifying this highly effective artemisinin for the treatment of malaria. And so this medicine, this plant was known to possess some good antiviral and antibacterial effects. So, and then we looked at can we have this plant in Turkey? Does it grow in Turkey? So when we look at it and investigate it, there are 22 species grown in the Turkey, different regions, as you can see here, in Mediterranean region, Marmara region, West Black Sea, and the Central Black Sea, Central Anatolia, Eastern Black Sea. And there are 22 species, which is Artemisia vulgaris, and all the names that you can see, and different. And our was the number 8, Artemisia annua. And there are, of course, some literature background that I can give you. There are studies, you know, and Artemisia that's been, as I said a minute ago, has been investigated heavily. So, and also comparison of different contents of the Artemisia extracts has been studies. And other examples in Turkey also, there is a study in Turkey, determination of artemisinin in selected Artemisia annua in Turkey with the reverse HPLC. So with our study, what we did basically, we collected Artemisia annua plant and then diverse extraction with the Saxlet, the ultrasonic assisted maceration and extraction. We did 21 different solvent extraction. We determined the artemisinin content in all these 21 extraction. And after that, we went to in vitro analysis. And then we did the SARS-CoV-2 neutralization assay with the cell viability studies. And after that, we investigated how does it bind, or can we investigate, or with the comparison of the new molecules or the other molecules. And we docked artemisinin in the binding site of the SARS-CoV-2 main protease, which is 3CL. So the plant material, as I mentioned, was actually collected from Bursa Uludağ. And these are the plant, the visuals, as you can see. Uludağ is the, I don't know if you ever been in Turkey, was the mountain near Bursa. It's known with the ski places, very touristic place. So in the outskirts of that Uludağ, well known with a lot of endemic plants, and as well as this artemisinin or Artemisia annua also is grown there. And these are the extraction studies, which I don't want to bore you too much. So we did a lot of different extraction studies, and as you can see with this Saxlet isolation, and then after that filtration, and then LCMS studies was actually concluded. And these are in the literature, there are several actually extraction methods of the artemisinin annua. Toluene, hexane, petrolameter, chloroform, ethanol, DCM, propylene glycol metal ether, isopropanol, and the list goes on. So our results, and these are the preliminary results that I want to share with you guys. So as you can see, these are the authentic material, and these are the 21 different extraction methods that we actually can at least share it with you guys. So our idea was, can we probably calculate in LCMS by mass the yield, or which isolation method was the best for us. Of course, we purchased the authentic material from the Aldrich artemisinin. This is the NMR study of it. And then we compare with our extracts with the authentic material with the LCMS ESI mass spectrum analysis. Then we did some precursor ion and collision energy study, and then identify each fragments with the LCMS. And we did some LOD and LOT studies with the calibration chart. This was actually, these are all in the manuscripts. With this 21 amino acid, with these extraction methods, the best one was actually, as you can see here, these AA3 and AA4 and 7, which is highlighted here. Then after that, these 3 extraction that then we sent those for in vitro studies. These are the IC50 values of the artemisinin for used in Vero 8E6 cells and HEC and 293T cells. The IC50 values are highlighted here, as you can see. And then compare those results with the main protease activity. As you can see here, so there are some inhibition of the main protease, and this is how it's. I think it's considered to be effective for COVID-19. And we did some pseudovirus neutralization assay using again artemisinin, and these are the results in the control studies. If you can see clearly with the infection, so that artemisinin show not of course 100% potent. Of course, it shows some decent activity against COVID-19. And these are the results. Also, we looked at the immunomodulatory effects of the artemisinin and pro-inflammatory cytokine production with the LPS challenge. And then we investigated its top-talking pose of artemisinin, again for SARS-CoV-2 main protease 3CL. And these are the results. And again, we compare those activity with the known ligands, as you can see here, with the manidipine, canidipine, and lefonidipine. And artemisinin showed actually a somewhat decent binding affinity with our docking studies. And these are the results. It's right now 39 minutes. We, of course, we just don't do COVID-19 projects. We have several different other projects that to deal with discovery and development for cancer and cancer metabolism, CNS diseases, Parkinson, neurodegenerative diseases, Alzheimer, another one, diabetes is another my passion. Half of my patents actually related to diabetes drug development. Inflammatory disorders is another my passion. CVS is another one. Cardiovascular system diseases, rare diseases, SMA subtypes, these Parkinson disease subtypes, early onset. Of course, what we do drug discovery, hit identification, lead optimization, drug development, structure-activity relationship, pharmacokinetics studies and pharmacodynamic studies, and of course, ADME properties of the lead molecules. And these are the 9 projects that we completed, and actually 1 or 2 of them still ongoing. Some of them Parkinson drug development, disease drug development, some of them cancer, some of them anti-parasitic leishmaniasis, for example. Another one, NAFLD, non-alcoholic fatty liver disease, for example. We still try to collaborate with researchers worldwide. And this slide was for youngsters, for young researchers. And excuse me, but if there are some on the listeners or audiences who wants to be a part of our team, please feel free to send your CVs to my email is highlighted here, and send your CV. We can consider you in your PhD or also your postdoctoral studies. And we have several other collaborative studies, which I don't want to bore you too much. For example, we have another phase 2 study for SARS-CoV-2, which is this is the drug repurposing studies. Montelukast was the medicine for asthma. As you know, patent expired in 2009. So we found some good activity in this one. Right now, 52 patients are they tried without using favipiravir or favipiravir. They get the benefit using Montelukast, and they actually were treated. They were feeling much better. And this is the clinical trials list. This is also patented work that we did. Right now, the publication is on. We have the bioarchive. Anybody who's interested. Another one we have cryptochrome studies. We discovered a molecule amount to 6 million, which actually bioarchive just came in today from this. Another research, another patent in US. This was a cancer drug development with US researchers with Hassan in Georgia Cancer Center. This research was also published in Nature Communications as well as Oncogene. And another patent for Parkinson drug development. This is leishmaniasis drug development. Again, this is another one. So the list goes on. And I'm about to finish, Hina. so I don't want to bore you too much. This is Istanbul Bosporus. This is actually where Medipol is located. This is actually close proximity. This is when actually there was a construction. Taken picture from the aerial and very close to Bosphorus. This is the picture when the Medipol is right now. We are hosting about 25,000 people right now in Medipol. This is International School of Medicine, Center of Drug Discovery and Development. This is where we do most of our research. And before I close, of course, I always acknowledge my team, our Center of Drug Discovery and our Güzel Research Group. We have 3 assistant professors in our group right now, 5 postdocs, and 5, 6, 7 actually PhD students right now, 3 MS students. We have, of course, former group members that are right now, some of them assistant professors, some of them postdocs, and some of them actually went to industry. We have several actually undergrad students who actually participated in our research, and I sincerely thank to my group. And this was the Favicover team when we were establishing the for that drug development. And I always thank to our team. We always thank to our collaborators as well as the acknowledge the Istanbul Medipol University president right now, who became a Minister of Health, Vice Minister of Health right now, Professor Sabahattin Aydın, and Gürkan Hoca in Sabita, and our collaborators worldwide. We sincerely thank to them. I sincerely thank our also funding resources, Medipol Bremer, BAP, and the Tibitak, Sci Life, which is the Swedish company, COST, European agency, and I want to thank to inviting Comstech and ICBH for this invitation, and on always open to collaboration. As I said in the beginning of my lecture or talk, this is a team effort. Without a team effort, this cannot be successful. So let's partnership together in order to develop new molecules. And I want to thank you for your attention. So if there is a questions, right now I am at the 45-minute enough. So I think we have time for questions. Please feel free to ask, and thank you for your nice invitation again. I appreciate that.
Thank you so much, sir. It was certainly not boring at all. It was very exciting talk, as always, and it's always good to see the Bosphorus and your excellent, beautiful campus. So with this, I request Hazima to take the questions.
Thank you, Dr. Hina. And I would also like to thank Professor Güzel for his time. And it's always wonderful to hear him talk. And actually, I would like to say that your humbleness even comes across the screen as well, virtually too. So we have a question in the chat box. Let me read them out for you. How about the toxicity studies on the drug? What are the mechanism of action? Does it have direct effects on the inhibition of COVID-19 virus proliferation?
Is this asked for Favicover or Favipiravir or for artemisinin?
Okay, I think that is not mentioned in the question in itself. So if you have a general response, then you can do it. And I would
if
like
this is a Favicover or Favipiravir, as you know, Favipiravir mechanism of action is it's the RNA-dependent polymerase inhibitor. It's the blocking the RNA-dependent. So basically, it stops the proliferating of the virus. So that's how it basically acts. Artemisinin annua, there are some studies. Of course, the mechanism of action is still unknown, but with our studies, it binds to main protease 3CL. This is what we at least noticed. But there are in the anti-malaria studies, of course, artemisia is has been heavily worked in that area. So there are some binding studies as well. But again, mechanism of action is, I would say, truly right now it's unknown for
Right.
artemisinin. If the question is for artemisinin.
Okay, thank you. There is another question. Are there some randomized double-blind clinical trials on the natural herbal remedies which can back up this claim of treatment or prevention of COVID-19?
This is a good question, actually. I was just highlighting that there are some studies, even Artemisia right now, with the quercetin, artemisinin, and vitamin C. There is a phase 2 study for COVID-19 right now. If you guys are, I think the product name, I was searching it, Artiveda. The product name is Artiveda, artemisinin, vitamin C, and I think it has also curcumin, if I remember correct. So it's it's a cocktail of natural products. Right now, it's in phase 2. As I mentioned, there's a black cumin, black sativa is also in phase 2 study right now, approved by FDA. Actually, believe it or not, there are 3-4 Pakistani brothers, doctors in the Texas region. They established a pharma company. They approached me actually for the black cumin. They actually got the approval by the FDA for the COVID-19 studies. So we want to integrate this in the Turkey as well as in like with the 20 or 30 patients. So the question, basically bottom line, yes, Kazima, there are some natural products which are on clinical studies for COVID-19 treatment, if that is the question.
Yes, thank you. And moving on to next question, Ms. Myra Khalid is asking, what is the role of Janus kinase inhibitors in the treatment of coronavirus disease?
Janus kinase is, as far as I know, it's the cancer target. I am not aware of Janus kinase research for the treatment of COVID-19, but there are some kinase studies for other kinase studies. I know that for COVID-19 treatments, but for Janus kinase, I'm not aware of that study. If you guys kindly send me the link, I can investigate that way. At least we can teach each other. So I'm not aware of a Janus kinase for COVID-19, but I know it's a good hot target for cancer drug development.
Right. Thank you. We have another question from Mr. Hassan. He's asking, how would the products developed will be effective against the different variants of the COVID-19, like the Delta virus or the new variants?
Yeah, that's a good question also. This is actually live and learn. It's a trial and error, because even the vaccines, we are not sure whether it's going to be preventive or not. Even like BioNTech, Sinovac, or other vaccines with the Delta or the other variants right now, commonly as you know, these mutants, extremely contagious and spreads very fast. But that toll is, of course, questionable. Inshallah, we will have to reduced that toll. But is there an effective medicine for the COVID-19 or the SARS-CoV-2 variants? As far as I know, there is none. So, I mean, there are a lot of studies going on. When you look at the clinical trial govs, that website, I mean, when you just click on COVID-19, you will see a lot of hits. So just naturals and synthetic molecules are heavily investigated in clinic right now. But for variants, I mean, right now, I can say virus is much faster than us, because research is, as you know, is very slow. I mean, you need, for example, for clinical outcome, you need 5,000 patients, Kazima, right? At least for phase 3. So in order to reach that level, or in a comfort level, look, this medicine or this synthetic or natural, it doesn't matter, works for this variant. You need 5,000, what is it, statistically relevant data. Unfortunately, we don't have that yet, for that variant at least, as far as I know. So at this point, unfortunately, virus spreading or virus mutating much faster than that we can do research.
Right. Thank you very much, Professor Ghazel, for your time. And it was such a wonderful talk. And even there are comments saying impressive, Professor Ghazel. Thank you for the presentation.
I appreciate that.
So again, thank you very much. And I would also like to thank Dr. Hina for her continuous, support in this whole day today. And again, thank you to all our speakers who were there today. And with this, we come to end of our first day of the meeting. And with this, again, I would like to thank our participants for joining us today. We will begin our second day of the meeting tomorrow, inshallah, at 9:30 hours, Pakistan Standard Time. And with this, I would also like our participants to know that only participants who attend all the sessions of both days will get the certificates. So I will request you all to please join us tomorrow as well, and looking forward to have you all. And with this, over to Dr. Hina if she has something to say.
Nothing but just to thank Professor Ghazel for such an informative, exciting lecture and discussion as always. It's very good to have you on board. Thank you for accepting our invitation. Thank you, Fazima, for all the hard work and coordination. I think if we don't have Sir, do we have Sir with us?
No, he's actually in the meeting. I just checked with him.
Okay, so Professor Iqbal is in meeting, so I would like to thank you, Professor Ghazel, on his behalf as well, and looking forward to our fruitful collaboration.
Thank you.
Okay. Thank you very much.
Thank you, everyone. See you tomorrow.
Inshallah.
Thank you, guys.
Khuda Hafiz.
Khuda Hafiz.